Welcome to HHMI's 2016 Holiday Lectures on Science. This year's lectures, Ecology of Rivers and Coasts, Food Webs and Human Impacts, will be given by Dr. Mary Power of the University of California, Berkeley, and Dr. Brian Silliman of Duke University. The first lecture is titled Trophic Cascades in Rivers. And now, Dr. Mary Power.
So thanks so much for the Howard Hughes Medical Institute folks for inviting us to talk about ecology today. And the kind of ecology that we'll focus on is one that I'm particularly passionate about, food web ecology. So trophic cascades, this is a specialized term, but I hope it'll be vivid and clear for you soon. What happens to trophic cascades and what happens to them underwater in rivers. So ecology simply is the scientific study of how organisms interact with their environments, but these interactions are almost never simple and direct. They're almost always mediated through a web of indirect effects and other organisms and other factors that you may not be thinking about at the moment. So there's this complexity, and when we focus on the job that this little bug, this mayfly insect, an aquatic insect, has, it needs to go into this world, find enough to eat itself without becoming a prey. It has to eat and not be eaten. This is a big problem when you're in a dangerous, heterogeneous, changing world. So how do they do it? And how do we think about this and try to make predictions about whether the mayfly will be safe or the fish will get fed? We think about food webs, and food webs are always really oversimplified cartoons of nature, but this would be a characterization of how energy and nutrients flow up from the producers, the plants and the microscopic algae and seaweeds that fix carbon into edible forms by using sunlight energy. And then that's grazed by the herbivores, and the herbivores are eaten by predators, and some predators eat other predators. So here you have a depiction, kind of a roadmap of how energy and nutrients are flowing up through a web of connections. And we call these things trophic levels, the predators, the herbivores, the producers. Trophic is just a Greek word that means feeding. So they depict that, but they also depict something else, something rather different. And that is whether any of those predators can actually reduce the abundance of their prey, whether they can control populations. So can that heron really suppress the populations of the minnow or the steelhead fish there? Or if they were in abundance, could they suppress the insects? Or could the insects suppress the blooms of the algae? So we'll talk about that. And these are top-down effects. And if you find these chains through the complex web, you find some chains that are really strong and do mediate these chains of predators that can actually even ultimately affect plant abundance by their effects on their own prey, the prey's prey. So you find these bottom-moving, top-down controls, and that's what we call a trophic cascade, if that happens. So we're interested in that. Now, it's very natural for all of us to think, for example, where the world is green with a nice forest in Michigan, or where it's more barren-looking in dry prairies of Kansas. It's the first thing we think of, oh, it's got to be climate, that the plants have more water so we can grow forests in Michigan, and Kansas not so much. But it isn't necessarily what's controlling the vegetation. The thing that's hidden is the possibility that predators may be involved. And Michigan ecologists actually looked out their window, it looked green, so they said, well, maybe the world is green because predators are suppressing herbivores, the yellow circle, and herbivores then cannot suppress plants, so the predators are indirectly protecting the plants. So that would be a trophic cascade with 3 trophic levels. Now, a Kansas ecologist, Fretwell, looked out of his window, and it didn't look all that green in Kansas. So he said, well, maybe the world doesn't have to have 3 levels. What if it had fewer or more? So what he reasoned was an extension of this idea that if plants were alone in the world, one trophic level, they would probably grow up to an abundance where they eventually became limited by their own resource, water, nutrients, maybe sunlight. But if you added grazers, the plants would be suppressed and they wouldn't be resource limited anymore, they'd be grazer limited. And the grazers would be resource limited. They'd run out of plant food. But then you could add predators. You get the Michigan situation where the predators are protecting the plants and suppressing the grazers. And there can be important predators of predators. You'll hear about some in my talk. So you can get a situation where you have 4 trophic levels, or maybe 5, maybe even 6 sometimes. But there's a generalization that comes out of this idea. And that is that if the world has odd numbers of trophic levels, 1, 3, 5, it should look green. We should have vegetation released. And if it has even numbers, it would be barren with 2 or 4 with the grazers released to suppress the vegetation. And what happens to the vegetation is what happens to the world. Often vegetation structures the way we look and feel and can move through the world. It's huge. So a lot of the targets of this trophic cascade ecology involve trying to predict what is going to be the impact on plant biomass. So are some of you skeptical scientists or scientists-to-be already thinking about what's wrong with this really simple cartoonish idea? I see it, good. Okay, so there are things wrong. Plants, green doesn't mean edible. So that means that if we see a forest, it's a 1 trophic level system because the plants figured out how to be toxic or spiny or just not nutritious enough. Now, also, how do you count trophic levels when you have things like frogs that are very impolite and they hop from 1 trophic level to another? They're herbivores as babies, and then they grow and eat insects, and they're predators as adults. And we and rats and lots of other common species are also very omnivorous. So that's a problem. Then, if you're out there in the real world, you might be both hungry and afraid, and you're making trade-off decisions. Prey populations are often co-limited by fear and hunger, and those factors interact. You might make different decisions about foraging. So how can we say that the one population is always resource limited, another population is always consumer limited? So there are all these problems, and then there's a big problem that what limits life on Earth isn't always trophic. It isn't always getting enough food or being afraid of being eaten. We have other factors that can kill and change distributions. So you'd think with all of this going on in the real world, this theory would never work. But in my experience, it's been really useful. All theories are wrong, but some are useful, right? It's been really useful underwater to think about this model and try to understand then why is the world looking green, barren, why are populations thriving or not thriving. So let me tell you why I like working in rivers. Rivers, for one thing, are very convenient for experimental and comparative work because nature has offered you a string of beads where the beads might be replicated pool habitats that are connected by shallow riffles. So the dark blue in the drawing here are the deeper pools, habitats for bigger organisms. The riffles between are shallow and faster, and little organisms, or maybe the juveniles, would be there, but it would be dangerous for adults to be there for reasons I'll tell about later. You can imagine that they're victimized by terrestrial or bird predators. So that's one advantage in rivers, and another advantage is that life is miniaturized there. So when Sean and his partners are viewing grazing systems in the Serengeti, they have to charter an airplane to really get a good count of those animals, but I can just sit on a stream bank. My father actually helped me build my first experimental stream enclosure here in Panama, and Dad and I put that fence in, and there's a herd of armored catfish that you'll meet shortly grazing there, so I can keep track of what they're doing, and what they're grazing is also miniaturized. In that drawing, it's what you would perceive macroscopically as slime on a rock. But if you scrape it up and look under a microscope, it's a very diverse and beautiful world, and often it's thinner than the thickness of your hair. It's that little skin of life, but it's very, very interesting life, algae. Okay, so we're going to talk about two extremely different rivers and two extremely different grazers, grazing fish. Both eat algae. This first talk is going to emphasize how the traits of these grazers are making a difference in the length of food chains through food webs. So we'll start in Panama, where that big lumbering armored catfish is grazing. And before I change slides, just have a look. He's the guy in the middle. It's the most common bristlenose catfish in pools. And would you like to be a predator taking that fish head on? It would be uncomfortable, right? So here's the Rio Frijoles, Bean River of central Panama. And you're walking down this river. You come to the shallow riffle where the water's wrinkled and sparkly. And then you come to the deeper, quiet pool beyond. Then beyond, you can see the next riffle, and then there's another pool. And this would be in a sunny reach, where the river has kept a floodplain open. But you can also go to places where the rock is harder and the forest can grow right up to the edge of the river and shade the pools very darkly. So this repeated pool riffle sequence gives me a chance to compare what's going on in sunny pools and dark pools, which probably makes some sort of difference. You don't necessarily know what when you're first exploring. But it probably matters somehow to these grazing armored catfish and the algae, which is their food. So here they are. There were 4 species. Because they had armor, I could use just surgical wire and moccasin beads to mark them as individuals. I followed 1,300 fish for 3 years in my life with catfish. The most common is the black species, and it's called in the aquarium trade the bristlenose catfish. In deeper pools, this guy is absolutely safe because it's outgrown all the gape-limited predators. In the foreground, you can see there's a kerosene fish. This is the same family that gives us piranhas. And even these little guys are 24-hour-a-day active predators. They'll eat animals they can fit in their mouths, but they can't fit that big old armor catfish into their mouth. Now, what are the catfish doing with their mouths? Well, they're making kind of a seal over flat substrates with those suctorial discs, and they're using 4 comb-like tooth plates to scrape the algae off. These armored catfish, how many of you have seen them before? You can see 4 in aquaria out in the lobby, but people keep them in aquaria to keep the glass clean of algae. So they're very useful in aquaria. But what they're doing in nature is about the same thing. They're scraping algae off of flat surfaces. That makes it easier for an ecologist to know how much food is available to them day by day in their habitats. You can measure the area they can graze. You can measure the growth rate of algae on small parts of that area and extrapolate, and you've got how much food is renewing for a fish in a pool at a given time. And I just want to introduce you, you'll hear a lot more about algae in the 3rd lecture, to how cool it is. These are algae that grow on microscopic surfaces. That surface in the scanning electron micrograph in the right-hand side at top, it's growing on a surface that is the diameter of your human hair, okay? So that's what you have if you're an alga and you're getting grown upon. But those cells are really high-quality, nutritious food, and they grow very fast. That's important. But you can't really see anything but a thin, barely slippery skin if you and the armored catfish are competing for finding algae, because they'll win. So what you do is an experiment, very simple, but you just lift natural rocks or artificial tiles with the rock-like surface up above the water where the catfish can't get them. And then you can measure how fast the algae grow when they're not grazed in sunny and dark pools. And no surprise to those of you who know plants, the algae are growing a lot faster in the sunny pools. Those are the orange line, 2 pools. What I'm showing here is days, and the 16 days total. Over the first 4 days, the growth is really fast, and then it's starting to level off. So I look at the first 4 days, because the catfish don't give them time to level off in their growth. And that's the standing crop, or the abundance of algae that you'd measure just by taking a scrape. So it's like a snapshot of abundance, the mass per unit area, okay? So you can see that is growing faster in the 2 pools that are sunny than in the purple lines, which show results from the 2 shaded pools there. And here you can see the tiles that I used and set up in all of those pools. Now, the catfish are also more abundant in the sunny pools. This is the months of the year going from September to August. And here is the density, the number of individual catfish per square meter. So what we have in orange here are averages from about 4 sunny pools that I tracked them over a year. And what's this big change in density doing? You know, this is a bit counterintuitive for those of us who live on Earth where the area of our environment doesn't change like this. But in rivers, rivers expand when the rains come and they contract when the river dries out in the dry season. So all we're seeing here is the beginning of the dry season, and the density's getting higher because the catfish have less and less area, but then the rains come and they can expand out. And so that's what's happening in the rainy season. So we see the catfish are tracking density because they're a lot denser always in the sunny pools than in the dark pools. And those were data I got by getting on my mask and snorkel and snorkeling through all those pools with an underwater flashlight so I could look under the ledges. When I got in, a lot of the catfish were scared under the rocks, but I could find them. I could see it to the end of their little cavern refuges and just count them. So that worked well. But still, I didn't know if my snorkel counts were giving me information about where the fish were actually feeding or if they just hung out in those pools, maybe hid from me there, and then grazed elsewhere. So if you want to really know what's going on with their food renewal and their tracking or their feeding on it, you need to actually make direct observations of the fish in nature. So I watched 2 dark and 2 sunny pools just sitting on the riverbank for actually day and night, for days and days, because the fish are active day and night. At night I used dim underwater flashlights underwater and kept count of, here's some of my little marked armored catfish. So I would, every 15 minutes, I'd count the number of catfish out on this platform, which I'd gridded with little markers beforehand. And then between what we call those scan samples, where I got counts of how many fish were using the platform, I'd watch individual fishes of various species and sizes, keeping track by mapping it of how much grazing area they covered. So that little guy, maybe in my 5-minute observation, had covered this pink area. So I can interpolate from this 25 by 25 centimeter grid how much area it grazed on its own in that time. So I've got 2 kinds of information: counts of all the fish and grazing, individual grazing rates for different kinds of individual fish. And those 2 pieces of information let me compute how long it will be before any fish gets back to regraze a small site on that substrate. So this may be not totally intuitive, and we can discuss it maybe this afternoon, but if you want to make that estimate of the grazing pressure on the algae or the return time of any fish to graze that small site again, then that time is equal to the area that they're sharing together times the number of individuals that are using that area collectively times the area that an individual can graze per time. And if you just cancel area with area, individuals with individuals, you see that you get 1 over 1 over time on the right-hand side, which is equal to return time of the fish to a site on the left-hand side. So why do I care about this? Return time is also the time the algae have to grow between grazing bouts on average. So that's why it's important, because you can tell how much food is going to be at that small site when they come back if you know how fast the algae is growing and how long it had to grow before it got grazed again. So the return times in these pools with the dense populations of armored catfish are on the order of just half a day. That's not much time for growth. Very intensely grazed. And in the darker pools, they have between 1 and 5 days to graze, so a lot longer. But the algae are growing a lot faster in the sunny pools than in the dark pools. So the growth rates of the algae here are on the order of 5 or 6 or sometimes even 10 times greater than the growth rates in the dark pools. So multiply the rate at which algae can grow, mass per time, times time, and you get the mass, the algal standing crop that should be on that site when it's about to be regrazed. And this might be something that would take me a little bit of time with pencil and paper to understand, to check if I believed it. But if you're interested, you should check and see if you believe it. And if you don't believe it, tell me. It may be wrong, right? Okay. So this is the predicted algal biomass that should be at those sites in the sunny pools and in the dark pools. And the important thing to notice with these 4 numbers is that the estimates for the dark pools bracket the values for all the pools. So that suggests that the catfish are evening out the effects of more sun, which does let algae grow faster, but it gets grazed faster. So the catfish are grazing it more intensely, and that dampens out the effect of that added fast growth on the accumulation of algal biomass. Okay, so that's kind of interesting, and one can check that directly. These algal standing crops were really thin. They're barely slippery. So I had to take my Swiss Army knife out, flake off bedrock, take it under the microscope, boil off some of the algae with acid and count it. But that did the trick because I could see that the abundance of algae on the sunny pool substrates was about the same as the directly measured abundance of algae in the dark pools, which tests this prediction from the model. So that's what we've got. Here's a summary just of what I saw. This is actually averaged over 12 months and 16 pools that range in canopy from very dark with just a 2 or 3% open sky through the forest canopy over the pool to very sunny where most of the sky over the river would be open. So that's the sunny pool. So yes, algae are growing faster, productivity is higher in the sunny pools. The catfish density is tracking that, denser in the sunny pools. The algal biomass, the measured algal biomass, is the same in dark, half-shaded and sunny pools. And so then is the growth rate of these catfish. So the guys in the dark pool are growing just as fast as the guys in the sunny pool, even though their food is growing more slowly, but they're not dealing with the crowds. So this is what leads to the support for a very useful, very simple theory called the ideal free distribution. You don't normally think about catfish as being ideal and free, but now you really should. Because ideal means they are smart enough to make really informed decisions about which pool is the best for me at any time. They've got to know what the range of algal availability is in dark but uncrowded pools or sunny, productive but crowded pools. And they have to make a decision, would I be better off joining the crowd or would I be better off going to a more sparsely populated, darker pool because it's not as crowded and there might be leftover food there. So they're ideal because they're smart and also they're free. They can choose the best pool available in the whole string of beads, the whole 3 kilometers with 30 pools. They can choose the best at any time to make the most food available to them. And so that is not always a common situation because if there were a bully catfish or a bully any other kind of fish in a pool, they might not be able to join. But because they're big, armored, spiny, and tough, they can have their way. They can go where they want. And also, they might not be able to get through riffles, but they do when they're motivated to track this food. So this is what we see from pool to pool to pool to pool. Now, we're going to see a problem for these fish on a different spatial scale, which is when you go from the deep parts of the pool up towards the water's edge along a depth gradient. Suddenly, they're not so free anymore. Okay, so why is that? And here's the water's edge. Here's heavily grazed substrate where it's deeper. This green line happens right about at the 20 centimeter, that's about that deep, contour. The 20 centimeter contour, you start seeing a bathtub ring of algae in many places. Panama and other places grazing fish are common. And it's because something is keeping those grazers out of the shallow water, even when they're really hungry in the deeper water. So what's inhibiting them? Predation. These catfish can outgrow all the swimming predators because swimming predators are gape limited. They can only eat what they can fit into their mouths in general. But the aerial and terrestrial predators have claws and fangs, and they can tear their prey apart. So they have no problem taking an armored catfish and catching it here and then ripping through that armor, getting to the soft, unarmored belly, and eating the poor catfish. So these catfish are really conservative about their safety, and they let that algae go even when they're starving here. The time, of course, that they get it is when the river rises in the rainy season. Suddenly it's safe enough, but only for a while, okay? So that's what the situation is. You can go into many places of nature in the natural world and put your finger on a point in the landscape where the food web changes length. That's what we're seeing here. What we're seeing is a 2 trophic level world that's barren in the deeper water. Put your finger on where the bathtub ring begins. That's a 3 trophic level world in the shallows, and I tested that by exposing caged fish to herons in shallow and deep water, and learned some other stuff about that experimentally. Now let's look at a very different fish in Oklahoma, the grazing minnow called the stone roller, Campostoma. And look how different it is than those armored catfish. This fish, unfortunately for it, is thin and soft, and it remains vulnerable to predators throughout its life. Now, it's a very common fish, despite being so vulnerable. It's really common throughout our Midwest. And I studied it in south central Oklahoma, right on the Texas border. So here's Briar Creek, where I spent 3 years. Briar Creek has these sunny pools that go through long, shallow riffles to get to the next pool upstream. And so it's this pool, riffle, replicated habitat. And some of these pools, walking along Briar Creek, were very barren, but others were filled with green algae. They were extremely striking in their algal abundance. I'd studied Panama before, so I wasn't used to seeing this algal contrast pool to pool, and the pools seemed otherwise kind of similar. But the barren pools had schools of the grazing minnow, Campostoma, and the green pools lacked the minnows entirely and had their predators, which were spotted and largemouth bass. Well, we mapped a length of 14 pools, pool, riffle, and kept track of whether the pool was green with bass or barren with minnows, or whether it had both bass and minnows in it. And the first time we did this in November, there was just one pool where the 2 species overlapped. It happened to have a wide, shallow tail where the minnows could get a little bit of cover from the bass. Then we did it again, and we kept seeing this pattern repeat date after date, that the minnows really didn't overlap with bass in very many of those 14 pools until we got a flood. And then the flood rearranged the fish in this river, and we got 4 overlaps. But those sorted themselves out one way or another. So here's a pool with minnows turning into a bass pool. And so the pattern was reestablished until the next flood, a lot of overlap, and then it was reestablished. So at the scale of the reach, there's this striking, we call it complementarity. If they're bass, there's green algae, no minnows. Minnows, barren, no algae. So you're probably starting to think, I hope, trophic cascades. But let's see what happens when we rearrange the fish ourselves instead of just letting floods do it. So we took a green bass pool, we got rid of all the bass, and we split that pool down the middle, and we added minnows just to one side. Here's what it looked like 5 weeks later. The side to which we added the minnows is this side on the lower part of the screen. It's grazed barren here. That's just barren Oklahoma limestone and silt. And here's the green algae that is actually in what we call a 1 trophic level system, because we took the bass out, but the algae now are free to graze because they don't have the grazers. So here are the data from that. We are going from 10th of September through mid-October here in time, and the average algal height is a quick and fairly decent way to measure algal standing crop if you want to do it quickly. You just throw transects across the stream, measure the height of the algae, then you have a relationship you've developed between algal height and algal weight, and you can use that in a number of ways to estimate algae quickly in nature. The algae started out high on the side to which we added the minnows in that green pool, but they grazed it down to fairly barren state within weeks. And the side that lacked the minnows actually bloomed. We got growth of algae here because we had a rain in late September that stirred up nutrients. So in a one trophic level system, the plants can respond to their nutrients. But in a two trophic level system, that pulse of enrichment just goes to the fish. The plants don't see it in their biomass, although they do grow faster. Okay, so that's what's going on. And then this purple line is a control pool where there was a naturally occurring school of Campostoma in the minnow, and we just followed it. And they could also suppress algae against that little pulse from the rainstorm, that little pulse of nutrients. Now, the other experiment, obviously, is to throw a bass into a minnow pool and see what happens. And we did that in a barren pool. We just threw one bass in. And minnows, I wanted to see the difference between what happened to algae, again, algal height, against the same timeline. But here are shallow records that were collected from less than 20 centimeters. Remember, that's the threshold for the bathtub ring. And here are records of the algal height from deeper areas. So what happened was when the bass was first introduced, the minnows moved into the shallows. They had avoided them somewhat before, so the algae was higher, but they got that algae while they were worrying about the bass. They were eating nervously, I guess. And then the algae in both shallow and deep substrates was released because the minnows disappeared pretty fast. And here's that same school of minnows that we didn't manipulate but just followed as a control to see how algae changed when we didn't do anything. Okay, so you can turn the barren world into a green world again within weeks by adding a predator. But what's that predator doing? Is it turning green because the bass eats up all the minnows? Or is there something the minnows can do about the situation? There is predator avoidance. And to estimate how important that was, before we did these experiments, I fenced off the downstream and upstream riffles to see how many minnows were eaten and how many tried to escape to get to a safer world beyond the bass pool. And here they are. They're milling against this upstream fence, wishing they could get out, and they did eventually. We lifted the fence, but not before we counted them. So we know that of 74 initial minnows, 40 escaped and 34 were eaten. So here's a simple trophic cascade. Despite all those complexities that we talked about, it does seem to work in these systems. So you have Campostoma, when it's not inhibited by predators, can mow down algae, a barren world for 2 levels. If the bass are protecting the algae, it's a green world, and you don't see the Campostoma. So we've talked about 2 very different grazers, and it's their traits here that make the difference, a really striking difference. It's even more surprising because the Panamanian stream was really contrasting in how fast the algae could grow, because it went through open sunny areas and very darkly forested areas where the jungle canopy was pretty thick. Nevertheless, the algae looked uniformly scant throughout that, and it's because this is a grazer that has figured out how to be ideal and free, not get eaten. It's not threatened by predators as long as it stays deep enough. Now, in Briar Creek, the Oklahoma stream was uniformly sunny, very productive, yet the standing crops of algae, their abundance, were strikingly different in green and barren pools. And it's because this poor, thin, soft fish is not free to go eat in a bass pool, much as it would like to. So that's what sets up a 3 trophic level cascade that is very similar to other cascades you can read about in terrestrial and especially marine systems. So that's what we have come up with finding a simple idea that actually is very useful under the river's surface. And now I want to just quickly go through why might this be useful at a larger context to society. I think you can make an argument about trophic cascades influencing what we can get out of our freshwaters in general. And the argument is simplistic, but it's a useful starting point for thinking about it. It runs like this. We want clean water, we want safe drinking water, clear water often, and we also really enjoy watching predators and wildlife and catching big fish. Okay, that's kind of a given. Now, we talked about these food webs, but we never talked about where the biomass was concentrated in the food webs. And we know as terrestrial organisms, we're kind of used to having the forest plant material really have most of the biomass, and we animals are a little bit small compared to that. But that's terrestrial. And what can happen underwater is you can have the opposite situation. Literally, in that lake I showed you, Clearwater Lake, you would go to the rocks, they'd be barely slippery. You'd turn over rocks, you'd see a few bugs. So a little bit of algae here. Here's the terrestrial system with a lot of maybe forest or even grasslands, fewer buffalo and fewer wolves, right? So here we are, barely slippery algae, thin film of algae is supporting somehow a higher biomass of grazing insects, and that's supporting an even higher biomass, sometimes of great big fish. So how can that be? It works this way, that the algae are thin but very, very productive, growing really fast. Algae can sometimes double every day, or even have slightly faster growth rates when they're really cranking. So that little thin skin is growing like gangbusters, and also many algae, like these diatoms, are really nutritious, as I was saying. They're excellent food. So they can support the growth of insects. Even the insects can be more abundant than the algae. These insects are reproducing maybe every few weeks. These are reproducing every day, right? And they're growing fast, but not as fast as these. So they can store some of that energy in their longer-lived bodies, and the fish are reproducing maybe every couple of years. So they're even longer lived. So you start getting all of this energy sustaining and stored in big, long-lived predators. And if you understand these rates, you can start to understand what's not easy to understand, how a trophic biomass pyramid can rest on its pointy little head, be inverted. Interesting, huh? Now we're getting services from this trophic pyramid as well that have to do with the clean water. The fish suppress the bugs suppress the algae, and the algae don't grow up and make the pool green, right? So that's a good thing too. So we're getting two great services from natural freshwaters in healthy ecosystems. But what are we often seeing? I bet some of you have seen this or been excluded from your favorite swimming pools because of harmful algal blooms, right? It's happening, I think, in almost every state, maybe not Alaska. But this is a common, all too common situation. This is a sample of water that was just dipped up from a lake in the Bay Area of California. And this is what we call a very harmful algal bloom. Not much animal life can live here because the algae at night will suck all the oxygen out of the water, and some of these algae are toxic, unfortunately. So we'll talk about that more in lecture 3. But this is what we call in freshwater a very bottom-heavy trophic pyramid, where almost all the biomass is in algae. And I can make an argument to you that this all depends on how long the food chains can be. So if we do a thought experiment, this is simplistic again, but do an armchair tour with me as we go to a city like Phoenix, Arizona, where we basically can say we have 0 trophic levels in the watersheds. The streams are all dry, and the watersheds have been overgrazed and it's arid, so there's no terrestrial vegetation that's really grown back. So there's a lot of smog in Phoenix, and they've had to shut down water wells on the outskirts in those arid watersheds because the residents are drinking toxic level of nitrates in their well water. Nitrate is something that, along with a lot of other stuff, can rain down from smog, and at high levels it can interfere with babies' respiration. So you get what you call blue baby syndrome if you have too high levels of nitrates in the wells, and this is happening in water wells in areas like this. So that's 0 trophic level, no plants to take up the algae. If we had plants, it would be better because the plants use nitrate as a nutrient. So they take it and use it to assimilate plant tissue, which is not usually toxic. And that's better, but if you have too many plants, you have that eutrophic mess that we talked about with the green pools. So you might want insects, aquatic insects, to graze that algae, but there might be too many insects, and a lot of these aquatic insects emerge into clouds, and sometimes these clouds are doing things to us that we don't like. So you might want fish that would eat the insects. That'd be a 3-level web, right? 3 trophic levels. And we'd be even happier if we had 4 trophic levels, and we'd be even happier if we had 5 trophic levels, as this famous ecologist Jim Estes, who studied the sea otter trophic cascade. And here he is just a few months ago. So he's enjoying products of a freshwater ecosystem that made it out to the ocean. And these, you can make this argument that if we can keep the food chains long, we can keep aquatic ecosystems healthy. And how do we do that? Well, that's the subject really of my 3rd lecture. We really need to think about our watersheds to keep these ecosystems in a state where we can support the great top predators that through trophic cascades can suppress lower trophic levels that can otherwise be problems for us. Thank you. So I'd love to hear questions. Yes.
My name is Eleanor. I'm from Blair High School.
My middle name is Eleanor. Unbelievable.
And so when you have the bass in the pools and you don't really have any minnows left, what do the bass eat?
Did you note, if you go back and look at the slide, look how hollow his belly is. He's a very hungry bass. So there are other fish, and some of them are a little harder for the bass to eat, so they last longer, like sunfish, are deep-bodied, have spines. So there are prey, but those bass are really hungry, and they're really ready to get at those minnows when they get rearranged. I think the reason they can hold on is the floods do rearrange them. Also, something about fish, they can starve a long time without dying. They can stay in the game. They just really shrink down, and that's how they make it. They can make their living on pulses that they get occasionally, more than we could. Yeah. Did you have a question?
Hello, my name is Nia Webb. I'm from Duke Ellington School of the Arts. I wanted to know, what can we do to sustain clean water for us to do our basic everyday needs like showering and drinking?
Well, this is going to be a topic for the 3rd lecture. I won't address all of those questions for all of the society, but the main thing is think about what we're adding to these aquatic ecosystems and what we're taking out. So in general, we are taking out water for our use. We have to be very vigilant about what we've taken and what remains and what's necessary. But we've taken water out. We sometimes take fish or big gravel materials for construction out. That changes the stream. And then we add heat. How do we add heat? We are taking forests away. We're warming the earth. Taking water away will make the shallower water warmer. That's important. We're adding fine sediments. We're adding strange chemicals. So if we can just control the sources that we're responsible for in the watersheds and figure out our extraction, and you'll see in California, my work is under the influence of marijuana. We're trying to grapple with the issue that marijuana gets thirsty just when the river needs the water most. But there are ways to do that by storing water at other times. So we can think about timing and what we're taking and what we're putting out. And it's a very big question. I'd love to talk with you at more length and hear your ideas.
Thank you.
Yeah. Yes, in the back. In the gray sweater? Yeah, gray jacket.
I'm Larry from the New School of Northern Virginia, and I was wondering how long it would normally take a bass to eat like 30 or so of the minnows.
That's an interesting question, but when we've done aquarium studies, and you can see a bass with 5 minnows poking out of its mouth, kind of looking like a tycoon with 5 cigars. It was so funny. So they are voracious. A fish, this is also a little counterintuitive, a fish can often eat a prey that's half its own length. Just gets it in, swallows it, pushes it through. And so these piscivorous fish could make short work of that. And I think we are seeing on the order of probably under 2 weeks it could finish off all those minnows.
Hi. Wait, is this working? Okay, cool.
Yeah.
My name's Isabella. I go to Latin Valley High School, and I had a question about your bass and minnow experiment. So when you like split the pool so that it was one side of bass and then one side of minnows, and then I remember you talked about how like they weren't eating at all. Do you think that that like affected your results when you mixed them together because of how starving they were, or would that pattern be prevalent in natural ecosystems as well?
Well, when we split the pool, we actually took the bass out. The bass had been keeping the pool green, but there weren't any bass when we added the minnows. So it was basically we took the bass out, added minnows to one side, didn't put bass back in the green side. So it went from 3 trophic levels to 1 trophic level, but that's still green. But in the pool where we did add the bass, it had started out 2 trophic levels, we added the 3rd, and that's where his question is relevant about how fast could they have eaten those minnows up, and the answer is really fast. Does that answer your question? Were you asking a slightly different question?
I think it does.
Okay, thanks.
Thank you.
Yes.
So my name is Bruin. I'm from Rockridge High School, and you had mentioned the concept of nitrates being put into the river ecosystems. If you added algae or any sort of plant introducing to take care of the nitrates, then how long do you think would it take for that ecosystem to stabilize?
Oh, okay. I'm not even sure you could call it stable, to be honest, because you'd get the algae taking up those nitrates, they'd bloom, and then they'd be limited by something else. Maybe they'd shade themselves, and they'd probably collapse and rot, and there would be cycles, but I'm not sure they'd get to what we'd think of as an equilibrium state. Sometimes you do see equilibrium, and they can be short-term or long-term, but it's a very important question. Also, when something accumulates in nature, often something else figures out how to eat it. And even if the algae are toxic to animals, they have their own enemies, the viruses. The phage might attack and kill. So great big algal blooms, not the world's most stable ecosystem. Yeah.
Welcome to HHMI's 2016 Holiday Lectures on Science. This year's lectures, Ecology of Rivers and Coasts, Food Webs and Human Impacts, will be given by Dr. Mary Power of the University of California, Berkeley, and Dr. Brian Silliman of Duke University. The second lecture is titled Untangling Salt Marsh Food Webs. And now, Dr. Brian Silliman.
Thank you very much. First, I'd like to begin by thanking the Howard Hughes Medical Institute for both the invitation and support in putting together these lectures, and to thank you, the students and the teachers, for engaging with us in this science process, not only in understanding the lectures and discussing that, but in the activities asked for. These interactions are really important for progress in learning in science. And I'd like to start out today with 2 take-home lessons that I think are really important from this lecture. I'm going to present a story of scientific discovery, sprinkle in a little bit drama because everything's not cut and dry in science. And that is that the power of experiments are really important in understanding cause and effect relationships no matter what fields you're in. In mine, it's helped to unravel strong top-down forces in salt marsh food webs, pinpoint those, and then use that understanding to help conserve these ecosystems. The second one is that science is fun, exciting, investigative, and creative. And when I was your age, I was looking for something like that in a career. And my arrow pointed to history, because when I was in those classes, there was a lot of debate, there was a lot of discussion, evidence. I liked science, but a lot of it was memorization. And it was not until my 4th year of college that I started to do science, investigative science, just not test. And I want to encourage all you guys to do that as soon as possible if you want to test out science, because that's where you get the creativity and the excitement, is coming up with your own recipe and your own question. In today's lecture, I've organized that around 4 different questions. What are coastal wetlands? So I'm going to begin with a natural history overview. What organisms, what are the plants and animals that live there? How do coastal wetlands benefit humans? What are the services they provide us? And what controls coastal wetlands? What are the ecological forces that do that? Coastal wetlands are renowned across the world as one of the most highly productive ecosystems. Very luxuriant in the amount of plant production that will come out of that ecosystem in 1 year. And they primarily develop in protected areas like you would find here in this embayment. And if you look across a gradient of wave stress, you see how important it is for the protection from those waves for these wetlands to emerge. Here along this rocky shore, this is where we have high wave stress in the environment. And in that system, there's so many waves that the sand and the mud can't settle out. As we decrease the amount of wave stress coming into the system, the sand that's been entrained in that water will fall out, and you get the formation of beaches. But there's still enough storms and wave action to rip up any vegetation if it starts to grow. In these areas where it's more like a bathtub, we really don't have waves and that are very common, all the sediment starts to fall out, and we get this fine, muddy, silty environment. And that's where these lush intertidal grasslands can start to develop into these areas. They still need that seawall of protection, though. Where do they get that? On the east coast of the United States, we don't have the mountains along the coast. What we do have are vacation spots, the barrier islands. And this is an effectively long natural seawall that extends from the Florida coast all the way up to the Jersey shoreline. And behind that, we have a wave-protected zone. And in those areas, we have development of sediments, intertidal grasslands, and they can be as expanses of 7 miles in width along those areas. They're bounded on this side by wave stress and on this side by freshwater inputs. That allows freshwater plants to come in and outcompete them, so they can't move further inland. On the polar expansion, they're limited by ice. During those winters, it'll grab that grass and rip it up, so they can't go too far to the north. And in the south, they're limited by competition by bullies, these trees that grow up in the intertidal zone in protected areas. So in the temperate zone in protected areas, we see these lush intertidal grasslands. And that's the ecosystem I'm going to talk about today. They're also bounded over sort of shorter spatial scales. And if we take a sliver of that salt marsh, we can see how the tide really influences what you find in those ecosystems. Here is the cross-section of the salt marsh, and here are the different elevations of flooding between a low and high tide in the environment. Here's the mean low tide, and here's the highest high spring tide in the environment. The plants can't go lower than mean low tide because they'll drown. And they can't go higher because, again, they meet up with a competitive bully. They can't go in the terrestrial environments. They're outcompeted by those trees, which can't go into here because of a salty environment. Those tides also create striking gradients that are predictable in the amount of stress the organisms experience in that system. And that stress or predictably vary along that tidal gradient. Coming down the tidal gradient in the lower portions of the marsh, we have increasing oxygen stress. There's lower amounts of oxygen in that soil simply because that soil is underwater for a longer period of time. It's difficult for the oxygen to percolate in those soils. So those animals need adaptations in plants for a low oxygen environment. In the higher reaches, we have higher salt stress. This is not necessarily intuitive, but what happens at the high levels is you only get flooded in those areas 1 or 2 times a year. Brings in salt water, and over the period in between the flooding, we have evaporation, so you get concentration of salts. It's more like a desert environment in that. So we have increasing salt stress as we go up. Now, along these gradients, we also have striking zonation of the organisms in these environments. And what's so amazing about the salt marsh organisms, the plants and animals that live there, is you will not be overwhelmed by the diversity. It is not like a coral reef. And sometimes I wish I was in a coral reef because I want to see diversity. What you're overwhelmed with is the amount of organisms that fall into your boots. Okay? It's the sheer abundance that's overwhelming, especially when you go up to large spatial scales and think about how many organisms are there. So here are the plants, and up in this salty or desert-like environment, you find succulent plants like you would find in a desert. Those that are very conservative with their water budget. And then we find terrestrial-like plants on the border. Down in this low zone, we have grasses. And this looks like an agricultural field you find in the Midwest. These grasses are among the most productive ecosystems in the world. They rival that of tropical rainforests. They can grow to 9 to 10 feet tall in one growing season in the south. That's about this tall. It's like walking through a forest of grass. So very productive and abundant in those systems. The animals in the system, like the mollusk, are equally not as diverse but super abundant. In the snails, they occur in the higher intertidal system like this. These periwinkle snails like to climb up the stems of the plants as the tide comes in to avoid predators and continue to graze on the plants and the algae that's on them. And they come back down as the tide recedes to graze on the mudflat. These mussels occur in super high densities here underneath the plants and act like hotspots of, it's almost like a little spa. They keep more water there, there's less salt, they're like tree fertilizer stakes, they filter water, and they deposit feces right there and can enhance plant growth. These snails can occur 100 individuals per meter squared. Once you get a kilometer squared of marsh, it's 50 million snails, okay? And crabs will also occur in this abundance. Fiddler crabs are one of the most common crabs that we see in this environment, and they're very cool engineers. They dig holes in the marsh to avoid predators. When they come in, they go into those holes, and they just found out they plug them. They don't let the water in. They have a scuba tank of air in there because they breathe oxygen better in the air than they do underwater. That also enhances plant growth. Their densities can equal, if you scale it up to about 1/3 of an acre, the size of a typical suburban house lot, you're talking 150,000 crabs in your yard. And that means everybody in the neighborhood has 150,000 crabs. It's an amazing amount. We also have crabs that are predators. These are the blue crabs, one of my favorite things to eat. And these are predators that come in with the flood tide. They're swimming crabs. They can go over the marsh grass, and they come in, and they're scavenging, gathering crabs, eating them. They can eat a snail in 15 seconds. So they do a lot of consumption over a short period of time. Well, the food web doesn't stop there. We have our plants, we have the animals eating algae, and then we also have predators like this blue crabs, and we have predators above them. Here's some of those examples. We have sea otters that are just now expanding. This is very exciting, into salt marshes in California. Alligators that I found out 10 years ago occur in these marine systems and love to eat blue crabs. We have bonnethead sharks that are coming up co-occurring with climate change and the warming of waters in North Carolina. So all these animals are there. And if you multiply this vast abundance of organisms by the huge size of this ecosystem, there are over 4 million acres of salt marsh just in the United States. You get a generation of very important benefits and services for humans. One of those is near and dear to my heart. It's a model system for ecology. It doesn't serve as many people, but scientists. This is like a dollhouse for doing food web ecology because I can manipulate things that are small. I can change the level that they're on. I can change the number of them in small cages. It's not like working with a lion. A blue crab will pinch you and bite you and injure you a little bit, but it's not anything lethal. And the cages can be a lot smaller. So you can ask questions that are potentially generalizable that you can't, for instance, in a tree system. And the organisms are also short-lived. They also sequester carbon. We care about taking carbon out of the atmosphere. They're areas where we have high fish production. It enhances commercial as well as recreational fisheries. They protect our shorelines and they filter pollution. I'll explain these four in the next 4 slides. Carbon sequestration. There's a markets that are about ready just to open up where you can pay for carbon pollution, and the preservation of salt marshes we contend should be in that market. If you look at the amount of the carbon burial right here on the y-axis, on the x-axis are ecosystems. It's the log scale. What you see that salt marshes are about 10 to 20 times better at burying carbon over a period of time in comparison to forests, which we're most interested in this. And notice all your marine ecosystems are here. We call this blue carbon. And the reason that happens is because these plants are fixing carbon through photosynthesis. They're building roots, but because there's no oxygen or little oxygen in the ground, they don't decompose, and you get a peat layer that's growing over time. It's building up, and it's a carbon bank. They're also what I like to call food factories. Okay? These grasses are like big fences. They keep the predators out, and there's a lot of food. It's a good place for small organisms to hang out and grow fast. And with the expansion of salt marshes and greater area of salt marshes and areas, we see a strong correlation with increasing amount of important fisheries, small commercial fisheries like drum, shrimp, and blue crabs. And this data right here shows this positive linear relationship. With increasing area of the vegetated estuary, specifically salt marshes, we have a positive association with increasing number of shrimp that fishermen catch in their nets. It's really important because it boosts the local economy. What about shoreline protection? This is kind of hot off the presses. Over the 5 or 10 years, we're saying, can living shorelines, instead of putting up these bulkheads, protect our shorelines just as well under instances of moderate wave stress? And the answer is yes. We've done experiments. The marshes dampen the wave stress, and they keep the sediments in its place. That's very important if you're a local landholder and you don't want to lose your land in that environment. And when you use marshes and oysters instead of the seawall, you not only get protection, you get added benefits. You get fisheries protection. You get carbon sequestration. So it starts to add on. These living shorelines are an important service in these environments. They're also filters. They're sponges that suck up pollution that is especially coming into these estuaries where we have a lot of human development and runoff. And some of those pollutants that come in there include nutrients, nitrogen and phosphorus that you use, many people use in their yard. A lot of that runs off into these estuaries. There's also heavy metals, for instance. As they pass from the urban environment, before they get into the estuary, they have to go over all these salt marshes. And those plants take up those nutrients, they fix it in organic carbon, they can put it in a food web, and they can lock down some of those heavy metals. So important services like that. So this may have you begging the question, I started earlier, this is a very stressful system with few organisms, yet has the paradox of being super productive. How is that the case? So we asked this question, and it's something that salt marsh scientists have been studying for a long time. And one of the hypotheses they've looked at, and actually has a strong support, is that for those organisms that have adapted to that stress, there's a huge bonus. It's called the tidal subsidy. And this tidal subsidy, this is taken from one of my favorite places on earth, Sapelo Island, Georgia. See this luxuriant production I was talking about before? This is the grass in Georgia that gets about 9 feet tall by the end of the growing season. Really expansive ecosystem here. Oysters along the creek bank. This is a picture at low tide. They have 3.5 meter tides. And when that tide starts to come in, it's bringing oxygenated water, okay? And that's bringing oxygen percolating down to the roots. It's bringing in more plankton for the filter feeders. And it's taking away some of the toxins that can start to build up, the sulfides that give it that egg smell. And we have found that with increasing amount of flushing, not surprisingly, you have increasing what? Increasing amount of grass production. So there is a strong tidal subsidy to the environment. And we've also looked at other physical factors that could control marsh production, because these salt marshes are built on the foundation of plants. We call them foundation species. Okay? So how they go, the ecosystem goes. So as ecologists, we're interested, that's a focal species. It's creating the ecosystem. What are the factors that control its success? And we found a variety of physical factors are very important. I've talked about salt and oxygen. One that's really important is nutrients. And we found by taking plots, sprinkling nitrogen on versus phosphorus, that this system is very nitrogen in need. It's nitrogen limited. If you sprinkle nitrogen on there, the grass really grows, just like your backyard. So it's limited by those nutrients. The sediment type, river inputs, and all these other physical factors can regulate how much nitrogen is in that system. And combined, ecologists, we call these examples of bottom-up factors that can regulate plant growth. Nutrients and nutrient-regulating factors. And those nutrients can have a positive effect on plants and resource availability. And if you put this into a very simple conceptual model, ecological model, here we have our plants are our primary producer in the system, and the amount of nutrients and water and oxygen available to those plants will be a good predictor of how many plants and biomass and population of those plants you have in that system. But we also know that food webs are more complex than this. Plants many times get eaten. And so that can be controlled also from the top down. This is a dramatic example of top-down control. This this elephant has ignored the grassland, is going for the good stuff in the trees. They're knocking down that tree and then eating the limbs. That's a lethal interaction with the herbivore. It's killing that plant. And these elephants can have a strong effect on how many plants are in the environment. And we call that top-down control. That's a negative interaction right there. It's making that box even smaller. If we overlay those together, we're starting getting a conceptual model looking at the relative effects of bottom-up and top-down forces. We have a plant in our system limited by bottom-up as well as top-down effects. And of course, like plants, herbivores can also be limited by the amount of things, their resources, the food that they're bringing into the environment. In these systems, we also have 3 levels in many cases, and that's a predator. We can add to a predator system. And then, like plants, herbivores can be limited either from the top or the bottom up in these systems, and predators can exert strong top-down control on herbivores in certain situations. A good example of this 3-level trophic system, trophic meaning feeding, comes to us from the Midwestern part of the United States in terrestrial systems. And there we have wolves that exert top-down control on elks by eating them and also scaring them. It suppresses the reproduction rate. And the elks control the plant community by eating lots of saplings, and they suppress forests from coming back. Now, right now, I'd like for you to just concentrate on top-down control. The blue arrows here. And let's look at an example from a marine system, okay, a very similar system, and let's try to predict the dynamics that occur when we have double negative interactions. So again, here's one example that we have from the kelp system, one of my favorite examples. Sea otters eat urchins and control their densities. Urchins at high densities control the luxuriant production you see in kelp beds. They can form fronts and kill the entire ecosystem. So there's strong top-down control in the system. And what we had found, what do you think is going to happen when you have 2 negative interactions? What does that usually add up to? A positive or negative? Right. So we get a positive, and this is a facilitation. And it actually turned out to be the case. When we killed off the sea otters for their pelts 100 years ago, we saw urchin populations increase, and they decimated kelp populations. When the Endangered Species Act came into fruition, the sea otters were brought back, and summarily, as the sea otters recolonized an area, the urchins went down in numbers, and we saw kelps recover. This is strong evidence for top-down control through what we call a trophic cascade. That is, 2 direct interactions lead to a cascading indirect effect in the system. In this case, it's facilitation. So if we take this model of top-down versus bottom-up control, let's apply it to what we know about coastal wetlands. Okay? Early studies in the 1950s and 60s went in there and said, is there top-down control in the system? They collected the grasshoppers, the snails, and they looked what was in their guts. And they found mostly dead plant material, and they saw that the plants didn't have many bites on it. From that observational data, they concluded that grazers were inconsequential in this environment. That instead, the plants had won out on the evolutionary arms race, so we can just eliminate that box. We don't even have to worry about food webs. Let's just worry about physical factors and bottom-up control in the system. But there's still a lot of animals. And they said they're getting their nutrition not from eating live plants, but from eating dead ones. And we call those detritivores. And the idea about this, how organisms get the nutrition they need, is that the plants are dying on their own terms. They're then infected, that dead material, by fungus and bacteria. And that's effectively a cheese and crackers, I like to call it. The cracker being the carbon from the dead plant, and the cheese being protein and fats, lipids that increase over time as fungus and bacteria build up populations there. And those are the goodies that these grazers are waiting for at the picnic table. Okay? So that theoretical understanding of the system, or idea, then gives us predictions about what happens when we find an ecosystem that's dying with lots of invertebrates. So this is very common in salt marshes. We have localized die-off areas where the grasses are dying back, they're dead, and there's lots of invertebrates in the areas. And they always assume these areas were detrital lollipops. Physical stress had come into the area, there's lack of nutrients, the plants are dying, there's a lot of cheese and crackers, and so grazers are moving into the area. We all know there's difficulty in correlation and causation because you can be mixing up your independent and dependent variables. And they never tested the alternative hypothesis with experiments that grazers could be generating these areas. When I was just a little bit older than you, I went out into the salt marshes and I saw these grazers not only eating the cheese and crackers, they were big densities, and they were grazing the grass. And they were doing it in a very unique way, not like a grasshopper. These snails I knew from high school biology had belts of teeth that come out of their mouth, like the movie Alien. And it's rows of teeth, and it comes out like this on a big stick, and it rafts away, and they can make razor blade-like cuts in the stems right here. And what was in there, it looked like it was infected with fungus. So based on these observations, I then asked the question, can snails that actually do graze live grass control the growth of the amount of biomass we see out there? And what is the relative effect of that top-down control in relation to adding, sprinkling more nutrients into the environment? So I got in my truck, went down to the hardware store, got some hardware cloth that has zinc on it, and the snails absolutely hate to climb on it. So it's a perfect contraption for building. So again, both top-down and bottom-up questions. But then I put cages out in the marsh. And I did a fully factorial experiment. We have a control situation, which is normal snail abundance, 100, 200 snails per meter squared. I removed the snails with normal nitrogen. I added up resources, added resources for the plants with more nitrogen, and this is normal snail abundance, and then I took away the snails and then added nitrogen, okay? And we got whopping effects here, both strong bottom-up and top-down effects. What we found here, this is what a marsh looks like at the end of season, okay? And this is a marsh on snails. A marsh off snails, you can see that there's almost a 60, 70% reduction in biomass just due to the natural course of grazing in these systems. So it's currently under strong top-down control, and that's counter to the theory of what I read in the books. If we cross that with nitrogen, we see that nitrogen indeed will increase production. It's resource limited. But the difference between control snails and that of removal is now it's a 6-fold increase. And that means that the top-down effect of snails is even stronger with more nutrients. You add nutrients to the marsh, it's going to stimulate these snails to graze, and there's actually more of those grazing marks, which I call radulations, because their teeth are radula on those stems when they were fertilized. They're likely much tastier. So those of you who don't believe pictures, here's the data. These are the treatments of snails. This is control snails, and if you remove them, and the amount of grass in that system. And orange is not fertilized, and blue is fertilized. And what you see in the not fertilized treatments is that if you remove snails, you get a 3-fold increase in biomass. However, you get a much stronger effect in an interaction. The effect of snail removal depends on the presence of nitrogen. You add more resources, and the effect of removing them, boom, goes way up. Those snails were keeping those plants from attaining the high biomass that they could. Their top-down effect is stronger under nitrogen in that system. So I presented this at scientific meetings, and scientists walked out of the room. They said, that is absolutely not happening. It must happen at night. I've never seen those snails graze that grass. There is no way that we're turning over the marsh paradigm based on this information. So what do? I was like thinking about what could reconcile this. They never found live plant material in the snails' guts in the environment, but I'm seeing them graze this grass. But it looks like they're grazing fungus. Maybe they're farming fungus, and we agree that's what they're after. They're just getting it in a different way, more active way. So I asked some of the scientists, didn't believe me. Let's do experiments to test our relative ideas. So first thing we did is we went out into the marsh and we formed this question. Are snails farming fungus on salt marsh gourd grass leaves? And does this facilitation of fungus, an infection in the plant, is the mechanism by which they're controlling growth? And if that's the case, it would reconcile our two observations that seem counterintuitive. So the first thing we did is we did a natural survey. This is observational work. And we went out to areas that were and sampled leaves that were unscarred and those that were scarred. And this is the amount of fungal biomass on those two types of leaves. And you can see there's a 1,500% increase in the amount of fungal biomass on leaves that have been scarred. So it looks like the snails' activity are facilitating some kind of infection in the plants. But we need to test that experimentally. We don't know if there's a causal relationship. So we did that. We had a natural condition, and then we removed snails, and then we removed snails and simulated their radulations with a little razor blade. It took a long time. Each one of those snail cuts was simulated in those grass blades. And what we see is experimental evidence that snails do facilitate fungus. If you remove them, the fungus disappears, the scars start to heal. And this was very interesting. If you take them away but you simulate, you get more fungal biomass. Fungal biomass is on the y-axis here. And this suggests that the snails are facilitating fungus and then cropping down what happens to be their favorite food in that environment, potentially benefited from it. How do the snails, so the fungus benefit from this interaction? There's more fungus when snails are present. How does that affect the snails in turn? So we went out there and took little baby snails, because they grow faster, and we gave them these different substrates and asked them how fast they would grow if these substrates affected their growth rates. Indeed, it did. And it was proportional to how much fungus was there. This is the amount of snail growth, and these are the same treatments here. And what you see with more fungus increasing fungus availability, we have increasing snail growth rate. They didn't die without it, but they just stopped growing very much. So we have 3 species in this interaction, though. There's a host. There's a plant. How does this interaction affect a plant? Well, you have to tease. We know that snails have a negative effect. Is the mechanism disease? You have to take that disease away. The way we do that is with ointments. So I used a fungus version of ointment, like a Neosporin. We just put a fungicide on the environment with snails. So we had 30 cages with snails, and 15 of them got fungicide. What is the effect on the plants? And it was a really amazing effect, I thought, is that both these plots have 150 snails, and after 4 months, when you apply fungicide, you simulate the effect of removing the snails in that environment. So this suggests that almost all the top-down effect that snails are exerting on the system is because they're facilitating disease, and we know that's because they're farming the fungus on the leaf surface. The snails in this environment didn't die, but they just slowed the growth over time, and they continued to graze the grass during that time period. So then this asks the question, these snails can control grass growth through facilitation of fungal infection. They can have a big top-down effect. What's stopping them from killing all these grasses? Well, anybody's been out and swam in a marsh? I don't know if anybody's snorkeled in a marsh. I have. It's one of my favorite things to do. Blue crabs are all over the place. They go in there, and they're grabbing crabs or grabbing snails. And so an intuitive hypothesis is that potentially these snail populations are limited by predators in the environment. And we asked whether or not a trophic... So, onto the heretical hypothesis, does a trophic cascade regulate salt marsh grasses where predators suppress high densities of snails and keep those grazers in check? And we tested the effects of predators on grazers in 2 different ways. We used one of my favorite techniques, tethering snails, and we looked at whether or not in areas where you had high crab density, do you also have high predation on snails and low snail density? And then we excluded the predators in this environment, which are primarily blue crabs. And we said, does the population of snails change over time? So we tethered about 2,000 or 3,000 snails. This takes about 4 or 5 days sitting on the beach. You take fishing line, you have glue, you take little PVC pipes, you make like clothespins, and then you have all these snails and hope they don't tangle up their lines. You spread them all out, and then you go out in the marsh and you go down the areas like this that are loaded with alligators, so you're looking over your shoulder looking out for alligators. And you plug those in the marshes in areas with both high and low densities of crabs, and look what happens after a tidal cycle, or 2 tidal cycles, or 3. And then you also put predator exclusion cages and come back like 4 or 5 months later. And this is what we found with tethering. 4 or 5 days of work, nonstop work, was destroyed by blue crabs in about 4 hours in these systems. This is snail mortality in high crab density areas. 98% of 1,000 snails that were laid out were cracked open. These snails weren't picked off by fish. They were cracked open, which is a telltale sign of a blue crab. Remember, they can eat one in 15 seconds. It's amazing to see one do that. They put it in their claws. They spin it around with their mouth parts. They pick out a weak spot. They clamp down. It breaks it, and they spin around like a corn on the cob and eat all of it. It's pretty amazing. So in low crab density areas, 1 snail was lost every other day. So this suggests that, and we also found very few snails in these areas, but high abundance of snails. This suggests it's observational data that predators can control their density. If you combine that with cage exclusion results and you keep those predators out over a 5-month period, the number of snails they're recruiting, the babies come in from the water column, they land on grasses, and their survivorship goes way up, and they start growing into adults. And so this experimentally demonstrates that predators can control their population. And if you put this together, this is the conceptual model we have. We've got to get rid of the red X. This is what I'm telling everybody in the audience. And they're like, they love the red X. We don't have to worry about the food webs. That snails are a grazer, experimentally demonstrating they have strong top-down control and that predators control the density. And the response I got is that it only happens in the cage world. If you put too many animals in there, you're going to get overgrazing. Is there a relationship? Is there evidence that's outside a cage? And so ecologists have to ask, does cage world equal real world? And we use other methods. And we start using correlational approaches because we have a mechanistic understanding that can give us some hypotheses. So I went to long-term data, Department of Natural Resources, monitored blue crabs, and looked at the relationship of blue crab variation, snail density variation, and the amount of marsh grass over a 15-year period. So just looking at the top of the food web, this is the variation in blue crab abundance from trawls by the Department of Natural Resources over a 15-year period. And here's snail density, and you see a negative relationship. In years that you have more blue crabs, you have lower snails. In years with lower blue crabs, you have higher densities of snails. And in long-term monitoring plots in those same areas where we're measuring snail density, we saw a negative relationship at the lower part of this trophic level between snails and grass. As you increase number of snails in the system, you lower the grass biomass. And this suggests that there is spatial and temporal generality of this phenomena that we're experimentally demonstrating in these grasses. So people started to jive about this, the alternative hypothesis, but what they said now is, we believe it's going in southeastern salt marshes. It's probably not going on elsewhere. And it's probably idiosyncratic just where those snails are distributed in Georgia and South Carolina. What this makes us do is that we have to get rid of that red X for this region, our food web and understanding here. And things change when that happens. Get rid of the red X, the herbivore box changes. We know snails are really important. That top-down arrow becomes prominent. The amount of detritivores in the system is less than we thought because we mischaracterized this organism. And indeed, we have indirect facilitative interactions going on in this system, in this environment. So but what about, is there generality? Is top-down control of marshes, like my critics are saying, just idiosyncratic? It's just you just found it there, it's part of the natural history. Or is it a process that's common around the world? So the past 15 years we worked with high school students, college students around the world. They set up experiments and they tested these ideas. And what we found was it wasn't unique to that spot. It wasn't novel. It was, in fact, very common. So we went to Argentina and Chile. We went to New Zealand, Australia, a lot in Europe, and we built a big network. And we excluded the grazers. And what we found, there was strong control of these plants by the grazer populations. Very interestingly, look at the diversity. The players were all changing in these systems, but the process was the same. And we saw one of the most abundant organisms that we saw were crabs. And actually, we should put that here. We just found that the crabs are here in California. So this is science that's happening right now. And it's going on, and students like you are involved. And these crabs, like the snails, facilitate fungus, so they can have a strong top-down effect in the system. So grazers are really strong in the system. Okay, let's think about predators. We've only done this in one other system, and it's unpublished. Okay, so this is also research you could get involved in if you were interested in these other systems. Now, sea otters, it turns out, are coming into salt marshes in California. I was super excited because when I grew up thinking about studying food webs in salt marshes, it was just about small organisms, and I had ecosystem envy. I saw the sea otter story in my textbook. I'm like, wow, that's amazing. You can have orcas and sea otters and sharks at the top. We don't have that. Well, yeah, we do. It's just our books were wrong. And it turns out when you conserve otters, they like to hang out in salt marshes. And the salt marshes in Elkhorn Slough, California, have been dying off, and people have attributed that, guess what, from physical factors, lack of sediment, climate change in those environments. If you go out there and look, we checked it out. They called me and said, what do you think about the processes in the salt marshes? So let's look and see if you have super abundant crabs on the creek bank, crabs stuffing plants in their mouth, eating during the daytime. And in this little pocket over here where the otters were starting to colonize, they were eating the crabs. And the mothers were out there snorkeling, eating the crabs. It was pretty amazing. So I said, I think we should probably test for the relative effects of top-down effects in the system. So with my collaborators, we tested this hypothesis, and it's not published, it's in the process. But here's some of the data. In areas where sea otters are coming in, we see lower crab abundance. And if you exclude the sea otters, check that out. You get a lot of holes. The crab, the larvae get in there, and they start to grow up as adults, and they start grazing down the plants. They start eating the roots. And on the creek bank, when this happens, you get a lot of erosion in those environments. So our idea here is the expansion of sea otters in here is reestablishing a trophic cascade, suppressing grazers, and likely going to rescue this system from the erosional process that everybody's worried about. It may not be physical processes. So here's the data again, which data is very important. Pictures. If they speak 1,000 words, you should put those in there too. We have sea otter exclusion and control. So exclude the otters, you get a threefold increase in crabs per trap. The number of burrows goes up, and we also see that with above ground biomass, the amount of plant goes up significantly, and that has increased over time as well. So if we put all this together, here's some conclusions that I'd like to make before we get to the questions. First is that top-down control by grazers and predators in coastal wetlands is an important factor. It controls the amount of plants in that system, and we know that that's linked to the amount of services that are being generated. More plants equals more fisheries production. And if you're interested in those services, we've got to incorporate food webs into the management. It's a new way of thinking about coastal wetlands. We have to redo our textbooks and redo the way we think about management. Management of blue crab populations and sea otter populations is important for the health of those ecosystems. We need to think about preserving those predators, and it may be even more important if other, like climate stressors, are imposed on those plants. Those predators protect them from overgrazing. Here's a more general conclusion, is that from the snails, we saw snails, really small biomass. It's not intuitive that they can control a massive, extensive ecosystem that's so vast. The idea is that small guys can have big effects in these ecosystems if they're interacting with microbial disease. And we're doing surveys, and we're finding this is the case in a lot of other ecosystems, and I'll talk about that in Lecture 3. And the last one is that trophic cascades occur across diverse ecosystems. Another paradigm that I was taught when I was about your age about trophic cascades is they can be very powerful, but they're very likely to happen in systems where plants are very tasty, like algae-based systems. Those plant systems that are woody and that have vascular plants are not susceptible. And this shows that they are very susceptible, and those food web interactions are a critical part. Understanding them is a critical part in understanding what controls the pyramid, the biomass pyramid, and also conserving those systems. Thank you very much. Question here.
Hello, my name is Nia. I'm from Duke Ellington School of the Arts. My question is, what can we do to promote people to know the benefits of of the marshes and the impacts that they have on society?
That's a very important question. So it's the job of scientists to do public speaking, actually write in blogs, and write in journals, actually in newspapers. Those are things that people read. The people that are managing these ecosystems and that care about the services, you don't read the journal articles that I'm producing. So there needs to be there's a science communication gap, and there's professionals. So working with places like the Howard Hughes Medical Institute, science communication, journalism departments, we're interacting with those now at the Duke Marine Lab to get the word out. It's also important to participate on panels, to volunteer time, and work with managers and conservationists. Go to those meetings. You have to get out of the silo and not just publish in those journal articles. You have to go participate in the public discussions. Great question. Yes?
Hi, I'm Eleanor. I go to Blair High School. I was wondering, so when you were talking about setting up the experiment with the crabs and the snails, you mentioned that there were alligators.
Yeah.
And so how do the alligators affect like the snail populations there and the crab populations and the grass populations?
Great question. You're thinking like a food web ecologist. First thing I had to figure out was like, why are alligators there? From Discovery Channel information, they don't have salt glands, so they shouldn't be in the marine environment. It turns out we've tracked them. We've captured over 200 of the alligators. We're looking at that question. Their guts are full of marine organisms, like 90% by weight. They eat shrimp. They have blue crabs. What they do about every 6 or 7 days is they go rest in freshwater environments, and they dump all their salts. Amazing adaptation for an organism to live there. So how could they be? They're eating blue crabs. Could they be affecting the trophic cascade? We've manipulated their numbers in swimming pools. Bit dangerous, but if you have the right people to do it and know what they're doing with handling alligators, you can do that. And they can affect small populations in swimming pools. My idea is that they're probably not, because they don't eat that much, controlling the blue crab populations. They're likely tracking them. However, they could scare blue crabs. Blue crabs are scared to death of these gators. If they smell them, up into the marsh. So what could happen is they could actually make the 3-level trophic cascade, instead of canceling out and suppressing blue crab effect on snails, they could amplify it because of the spatial complexity in the system. The alligators don't move into the marsh. They stay in the creeks. Great question. So we don't know. That's something you could help out with. Yeah. Over here.
Yeah, so you said the snails were like farming fungus on the grass. Like, how exactly does that work? Do they like actively cultivate the fungus on the grass so that they can consume it, or?
Right. Great question. And when I started to make these observations, I started to read about ant fungal farming interactions that I saw on the Discovery Channel. And they're very complex behaviors, where the ants will transfer cultivar down generations, purposely planted in areas. They will pick off competitors. They will add nutrients. If you look at the snails, and we have, in all those different ways, they don't show any of that complexity. And it turns out that fungal spores are everywhere. They're on the plant, so if you cut it with a razor blade, it gets infected. So simple mechanical opening facilitates the fungus. And I think simple positive feedbacks, because they cue it on the fungus, they'll continue to eat that. One of the interesting things about that is that they don't home to the scars. And that doesn't make sense evolutionarily. Why, if you're going to commit to something and put an effort into it, aren't you defending it? You're not going to be helping everybody else out. Well, it turns out the more these stems are scarred, they get shorter. And it increases their predation rate. And so they show absolutely no preference to going to one or the other. So it's characterized in the ant world, it's characterized as this it's the beginning of farming. It's simple mechanical behavior that facilitates the growth of your favorite food. So if you just threw oranges in your backyard because you knew they would grow, that would be considered farming at this low level. And then the idea is that in time, some organisms evolve more complex. Yeah. Oh, right here, sorry.
So in 2014, there was a really big die-off of aquatic life in the Pacific. It was called the Great Dying. What do you think caused that, specifically for sea otters? Because a lot of otters died. They were collecting hundreds of bodies every day.
So the die-off of organisms in the marine environment is a mystery. I'll talk about a mystery in lecture 3 and then you have to investigate the relative effects. I'm not familiar with the sea otter die-off, but I can tell you some of the hypotheses about sea otters disappearing has to do with either disease or predators in the environment. And one way they infer the relative importance of that is the number of sea otter bodies that turn up relative to the population drop. In Alaska, for instance, everybody saw sea otter populations dropping over time, and they thought it was disease. They only found about a handful of diseased organisms in the environment. However, they found a lot of killer whales, rogue killer whales, 3 or 4 male killer whales that were specializing in the sea otters. And when you do mathematical models, it told us in that situation that it was actually top-down control. That can vary depending on where you are. We need to investigate those. Up here.
My name is Sarah. I'm from Woodgrove High School. I was wondering if you've ever considered chitin, the natural polymer that's really abundant in crab shells and shrimp shells. Sorry. I was just wondering if you've ever considered that for the benefit to the plants in the marshes.
In the amount of chitin that's in the crabs and snails?
Yeah. So
in previous research. Nitrogen?
In previous research, chitin is beneficial to transpiration or as a natural insecticide or pesticide. So I was wondering if you've ever consider of that in.
I have not. That's an interesting idea. There's certainly feedbacks between the organisms that are not necessarily based on feeding in those environments. So the mussels that are in the environment that form, they do some feeding, and then they're defecating in the environment. A decomposition case, if there's exoskeletons that are being shedding, I have not looked and put in some of those skeletons in the environment. That's actually a pretty interesting idea that you could test, because the fiddler crabs are shedding their shells off at a fast and abundant enough rate to actually increase the amount of chitin in the area. They have direct effects of chitin. It also decomposes into nitrogen. That would be an interesting experiment to do, and easy enough. Cool question.
Thank you.
Come here. I'll make my way over.
I'm Larry from the New School of Northern Virginia, and I was wondering if the grasses, if they're dying off from from the fungal infections or from the snails eating the fungus out of them.
Well, both of them have to be there. And it turns out, if you use the experiments, is that the snails, the main mechanism is that they open up the wounds, and then the fungus gets in there, and the plants shift from a growth metabolism to a wound metabolism. And they continue to get that dead plant materials being generated, and the snails graze that, and it then reduces biomass over time. So it's a combination effect, but the fungus is incredibly important for that question. Okay.
Hi, I'm Jasmine. I go to Loudon Valley High School, and my question for you is, after coming to your conclusions, would you consider the effects of top-down forces to be greater than bottom-up forces?
I think both of those are an important system. If you remove bottom-up forces from the system, you won't have plants. So you do need nutrients for it to grow. And I think they're both very, very strong, and their relative effects can change depending on the consumers in the system and the amount of nutrients. So we can't ignore either one or the other. We have to investigate both.
Thank you.
Welcome to HHMI's 2016 Holiday Lectures on Science. This year's lectures, Ecology of Rivers and Coasts, Food Webs and Human Impacts, will be given by Dr. Mary Power of the University of California, Berkeley, and Dr. Brian Silliman of Duke University. The third lecture is titled Floods, Droughts, and Food Chains. And now, Dr. Mary Power.
Thanks a lot. So our first lecture emphasized how fish with really different traits in really different rivers could either control or not control algae. And a number of questions came up about how the environment controls these organisms and their interactions, and that's what we'll stress today. We'll talk about the same river ecosystem under very different environmental regimes, and the emphasis will be on how you can have the same cast of characters, but the rules within the food web that determine how long the chains will be, these trophic cascades, will vary from year to year. So the river we'll look at is one I've studied for 25 years now. Time passes fast when you're doing ecology. Here in Northwest California, it's the Eel River. The Eel is actually the third largest river in California. People don't tend to know it as well because it's not densely populated, not that much agriculture, forestry, but no agriculture up there. It covers about 10,000 square kilometers of this whole drainage basin, and it dumps into the Pacific Ocean. Of course, being in coastal California, it has what we call a Mediterranean climate. That's the winter wet, summer dry seasonality that the Mediterranean has, but also California and Australia, South Africa. So it's a very extreme seasonal difference. But we tend to have predictably dry summers, and that's when most of the biological activity occurs. The winter floods are still quite important. Of course, that's the high water that the salmon are using to home back from the ocean and get up to their spawning areas in the headwaters of these rivers. But the floods also do something very interesting to the bed of the river. They can mobilize it all. If you're watching cows graze a pasture, imagine if you saw an earthquake that churned the pasture up so the cows are thrown hoof over horns and just ground up and all the grass was buried. Well, that's what goes on when rivers scour their beds. The whole substrate mobilizes. Anything that was attached to those rocks is scraped off. And so you start after the scouring floods have happened the previous winter. In the early spring, you don't see much rock-bound biology on those stones. There's silt that's falling out here as the river is subsiding and warming up. So this is the dry season coming on. Water's getting clear, but not much life until a few months later, you get huge blooms of algae following winter floods. So here are boulders and bedrocks that didn't get as severely scoured, and there were little basal cells left that then grew vegetatively to lengths of several meters, sometimes 10 meters across the stage. I call this Rapunzel's hair, for those of you who remember that old fairy tale. The algae are thin. They are actually about the diameter of human hair, but they can get really long. They're branched. There's a lot going on in these algae, as we'll find out. But here's what the seasonality looks like. If the river is flowing, the algae stay green, healthy, keep growing. But in the slack water areas, when we get into July, it's getting warmer, and some of those algae are stressed in more stagnant environments. They detach, they form these floating mats. Then, in both the turfs, the attached algae, and the floating mats wither back, and they form this funny-looking, stringy, knotted remnant at the end of the summer. So this strange architecture, which we see many years, is because these algae are infested with a little insect that weaves itself a little cocoon or shelter. I call it a tuft. So I call this the tuft-weaving midge. Midge are actually fly larvae, and we'll see their life cycle soon. The mother midge, the adult midge, looks a little bit like a mosquito, same large taxonomic group, but these guys usually don't bite. And she finds a floating mat, a favorite habitat for laying her eggs. The eggs grow into larvae that increase in size, weave that little tuft shelter, poke their head out, graze algae, and eventually pupate and emerge back as winged adults from the water. So that's their life cycle. And when we see this change, we notice that the midge numbers explode when those floating mats form. They get really abundant emergences of midges. And then when we see the mats wither back, they're full of larval midges that are still living in them. But we don't know from observation alone whether the collapse of the algae is caused by the grazing midges or whether the midges just collapse because they lost their algal habitat. So you don't know which causes which without doing an experiment. And in this river, we also didn't have any idea whether fish were important. We didn't have those big algae grazing fishes I mentioned in the first lecture, but there are carnivorous fishes, the juvenile salmonids, this is a little steelhead, and a minnow that eats a lot of insects as well. So to look at the effect of fish, we built boat-shaped enclosures around the boulders that were covered when we started these experiments in June with long turfs of algae. That's where most of the fish hang out, in that cover, and that's where most of the invertebrates are as well. So these are the rich little islands in a gravel-bedded river. We've done these experiments 5 years between 89 and 97. And we start them by building the boats and locking them into the natural stream bed, and then electroshocking out all the fish. The shocking stuns the fish, but it doesn't harm them, so they just are temporarily kind of motionless, and then you can net them out and release them in the open river, and they swim away, they're fine. So here are the enclosures and other different enclosures, and you can see that they're built around these algal-covered rocks. Some of these enclosures we kept as fish-free exclosures, no fish, and others we added the minnows and the steelhead, both or neither, to those other enclosures. So when the fish were present, to my great surprise, I was not expecting strong fish effects in this system, but we saw the algae collapse in just, again, 5 or 6 weeks. The algae that had started out 40 to 60 centimeters collapsed down to a close mat that was nowhere more than 1 or 2 centimeters high, covering the rock. And if you look closely underwater here, you can see that it still has that knotted, stringy appearance, as if it's filled with midges. If you looked into a fish exclosure from the top, you'd see the algae has changed color. It's been overgrown by globular, kind of red-colored blobs, which turn out to be a different kind of algae. And if you go underwater, the algae that was bright green, grass green, has now become a dingy yellow. So what's gone on? Just at the larger scale, here's what the whole biomass did. When fish were absent, we maintained the biomass of cladophora stayed erect, and here it is being tugged up because this brown globular thing, which is an alga called nostoc, it became 3 orders of magnitude, 1,000-fold higher when fish were absent than when fish were present. So here's a colony that's filled with its own oxygen floating up to the surface. So the blue here have fish, very little algae, no fish, lots of algae. So what caused this? Well, to my surprise, the presence of fish was also accompanied by the presence of lots and lots of midges. There were many, many more of these tuft-weaving midges where fish were present. And this was a shock to me because, like Brian's tethering, I had done midges in bondage experiment. I'd taken cotton thread, tied it around the midge, and put the midge into surface or benthic surface or bottom habitats or in algae or in gravel, just to see where the river was dangerous, where it wasn't as dangerous, to measure predation on them. And the fish love eating them when they're wiggling around on a thread. But when they're in those algal tufts, the fish in our system can't recognize them as prey. There is another predator that can deal with those midges, though, and that's an odonate larvae and other small predators, but this one in particular is important and interesting. How many of you have seen damselflies flying over freshwater? This is the juvenile form, and it feeds as a larvae. It's a predator. It has a mask of teeth that it folds over its face, and then it peeks over the mask and studies what is going on in that tuft for 7 or 8 minutes until it detects a movement. And then it sends its jaws, they unhinge right from here. This is all jaws. Sends those jaws into the tuft, grabs the midge, and withdraws it in a surgical strike. It doesn't even disrupt the algae. So it's very hard to detect their predation unless you watch them for 7, 8, or 8 minutes. And not that many of us, even naturalists, are zen enough to watch an insect for 7 or 8 minutes until it does something. But we were motivated to do that because of this larger experiment, to see if there was a causal relationship between that predator and the fact that with fish, you didn't have so many of that predator, and you had midges, but without the fish, you had no midges and a lot of those predators, fewer midges. So yes, in fact, it is a 4-level trophic cascade, a stronger one than I ever anticipated seeing the first time I did these experiments. You have larger fish that can suppress a small predator that, if released, can suppress a very important herbivore that can bow down the algae and create a barren world like that. And in the absence of those larger fish predators, you have the damselfly nymphs that become very common. They're not common in the open river. They're getting eaten there too. They're not common when fish are enclosed, but when they're exclosed, these guys have cover from fish predation. Their numbers increase, they control the herbivore, and the algae are released. So that was very encouraging and exciting. So we went out and did a lot of these experiments in the next 2 years, and we got incredibly disappointing results. We couldn't replicate our finding at all. No matter whether fish were present or absent in all of these 24 enclosures, the algae collapsed down to detritus within 3 or 4 weeks. Fish didn't make any difference at all. So what do we do? What's changed? And we finally woke up and noticed that we had entered one of those multi-year California droughts. So when we did the experiments in '89, we'd had winter scouring floods, but we hadn't had them before the 1990 or the 1991 summer experiments. Both of those winters were drier. And what changed was that in the absence of flooding, huge numbers of a big armored caddisfly, which is an herbivore. So this is a grazing insect that crawls around lugging this armored case and mows down algae if it's abundant. After a flood, you might see 1 or a couple per square meter, but after a drought, you might see almost up to 100 per square meter. Their densities are much, much higher when they haven't been blasted out of the winter river by scouring floods. So this grazer then became the story. The algae would start to bloom as the water settled and cleared, but it would get bald patches gnawed into it by these herds, these grazing fronts of Dicosmicus, the big, big caddisflies. This is one of the world's biggest caddisflies, and you can actually make really cool earrings out of them after the insects emerge, so it's cruelty-free. So anyway, the caddisflies are grazing and turning the world barren, so then we did the right experiment for a drought year, which would be we're taking 24 cages and we're stocking them with the caddisfly or the steelhead fish, the predator, or both or neither. So here's steelhead and caddisflies together. Here both are absent. There's just the steelhead. Here there's just the caddisfly. So what's going on in these enclosures is that says that most of the impact on the algae during this drought is from the caddisfly, not the steelhead. Here's what the enclosures look like when Dicosmicus is present. Here are the data for algal biomass on the y-axis and no Dicosmicus here, Dicosmicus here. And I'm showing you two lines. The bottom line is steelhead are present. The top line is steelhead absent. And this top line shows that steelhead still have an effect, and it's still negative. There's more algae when steelhead are absent. But it's not nearly as strong as the direct two-level control by the big grazer. A big grazer which in our system can outgrow all the predators. There are just a few that can eat it. So when you have disturbance, flood disturbance that resets the food web, you get algae that get a chance to grow before the animals build up, and they become long and luxuriant. Then you get the midges and other grazers colonizing the algae. Then the insect predators and the fish predators come in as the river subsides and as the insects recover from the flooding and as the fish move down on their migration out to the sea, in the case of the steelhead. So you get a nice four-level food chain that supports fish and allows them to grow. But in this drought year, same characters are present, but these guys are a bit hungrier because the algae energy is all going up to be directly controlled by the second trophic level. They're getting this energy, so both the bottom-up flow of food to fish and the top-down control of fish indirectly to have a trophic cascade effect, those are both weak or non-existent following drought. Okay, so that's what the winter hydrology does, and here's what it looks like. We plot hydrographs or trace stories of what the water does over time against time. Here's October to October, that's the water year in a seasonal environment. And here's discharge, how much flow is moving through the river on the y-axis. And it's also biomass because these blue is water flow. Here's our scouring flood. It has to go past a threshold and then the bed all mobilizes. And then the river is settling down for the summer drought, which is the biologically productive period in Mediterranean ecosystems. And the algae get a chance to grow big, but then they're tracked by grazers. The grazers are tracked by predators. Now these guys are dropping towards late summer. The edible grazers are getting eaten or emerging, and the predators are either emerging if they're damselflies or migrating out to sea if they're salmon. And here come the invulnerable grazers. They took a long time to build up their biomass because instead of allocating growth, energy to growth, they're allocating it to building a stone case, using their silk to glue it together and lugging it around. So defended grazers are growing slower, but once they take over, once their numbers do build up, they're the top of the food web because they're defended. So that's why disturbance can knock these guys back and allow more food to flow up to predators. Disturbance in this sense, even though it has a negative connotation in English, is a really good thing for ecosystems. It kind of shakes and resets the tablecloth so predators can eat. So here's the drought hydrograph. We don't even get close to scouring floods in years like this. And the grazers have overwintered and now they're getting big, armored, and inedible, except that they're eating the algae and just keeping it barren and low. And we can also remove disturbance by impounding and artificially regulating water, and we've done that all over the world, but especially in California. So downstream from these dams, you tend to find lots of either armored grazers or grazers that glue little silk tents to rocks and then graze underneath them that are also allocating to defense and also slow to build up, but not edible once they take over. So here's the food web now, a fraction of the food web that we'll talk about next, because I want to tell you about an even scarier situation than not feeding salmon very well. And that is what happens when the summer drought is severely low. So this is where my work comes under the influence of marijuana and other factors. One of them is the multi-year California drought. And I want now to take you to the base of the food web and look at the wonders of algae. You've probably seen algae growing in gutters. You probably haven't realized that there's incredible adventure and beauty in that little slimy world down there, but there is. Algae can be thought of, in my view, as the good, the bad, and the structural. So the good are diatoms, and that's what those fish in both Oklahoma and Panama were eating, diatoms that grow on rocks. But diatoms can also grow on plants. We call them epiphytes if they do that. Epiphytes, epi means surface, like your epidermis is your surface skin. Phytes means plants. So things that grow on plants are called epiphytes. And so when that Rapunzel's hair gets going, it increases surface area a millionfold for things that attach to it, like diatoms. So this little package of the Cladophora, the green alga, buried by tasty diatoms is really good for the food web. Grazers love it. But these structural algae assemblages can also get overgrown by cyanobacteria, and I say bad in quotes. Every time we take a breath, we should thank cyanobacteria for putting oxygen in the air, right? But they also, unfortunately, and through no intention of theirs, some of them are quite toxic to humans and other vertebrates. So we have to be concerned about them when they proliferate in water. So let me take you through seasonality and show you what can happen in a severe drought. You start out in the early spring and late in early summer with Cladophora growing in a clean green state. It starts out, I'd like to say she has green hair in her youth. She becomes a blonde in middle age, and Rapunzel in her later age decides to be a redhead. And what this color really is thickening layers of diatoms. When Rapunzel turns to a blonde, she has a monolayer of diatoms that are just basically coating one layer over the surface filament shown here. But by the time she's a redhead, she's deeply, thickly encrusted with diatoms. And that's not good for the Cladophora, but it's great for the grazers, because these diatoms are the really nutritious things. If you had a handful of diatoms and sniffed it, you'd say, oh, they smell fishy, but it's really fish that smell diatomy, because all the healthy oils that we get from eating salmon, the polyunsaturated fatty acids, the PUFAs, and other really good things like carotenoids. Carrots are healthy because of the orange pigments. That's why diatoms are also orange or yellow. So all those things are great for us and for invertebrate and invertebrate grazers nutritionally. So the midge, our friend the midge that lives in this tuff, just loves eating diatoms. And if you look at midge poop, you see lots of empty diatom shells. Another neat thing about diatoms is they live within a glass case. And I do mean glass, as much silica dioxide as your great-grandmother's cut glass pickle dish. They use glass in a very unique way to make a little shell for themselves, but the shell has a lot of holes in it, so the midge enzymes can get the good stuff and just leach it out. And then they defecate out the empty cases, and we can tell what they're eating. Also, you could tell from, if you're a good natural historian, that when a midge settles into a rusty turf of Cladophora, it cleans up its neighborhood by grazing it green right around its tufts and its little stringy corridors here. So you can see that they're eating away the rusty diatoms and turning the algae green again close to home, and then look at their guts. So obviously they're loving it. But here's a way to measure quantitatively what it's doing for insect production. What Samantha, who's a Berkeley undergrad, is doing is setting up these emergence traps. You can take PVC pipe, cut windows in it so the temperature inside is the same as the temperature outside, and the windows have screens so insects can't get in and out. Then you put the trap so the top is just above the water surface, and you cover that with sticky transparent plastic with the sticky glue pointed down. So light gets through that plastic, the midges come and other insects come up to the surface, they pop out, they try to emerge, but unfortunately for them, they get caught and you get to count them and measure them. So you can tell how much bug meat is emerging from the river on rusty Cladophora, where there's 25 times more coming up, or yellow or green Cladophora, or other kinds of algae that are green and don't get these diatom epiphytes. So they're loving the rusty Cladophora and emerging. If the some of those midges were feeding aquatic predators, but if they emerge, it's still good because they're feeding bats, birds, spiders, lizards, and a very thriving riparian food web. Okay, so this food source that I'm talking about is happening here in summer, and it really depends on having low but enough flow. We do call it normally the summer drought period, but this is the summer severe drought period where the flow gets so low that in the sunny big pools, the water stops moving and gets warm and stagnant. And that's partly given to us by the California drought. People are debating how much forest cover is affecting that, but another serious issue, and please think about this in terms of what we do as a society about marijuana. A lot of it's from illegal burgeoning grows in the Emerald Triangle. Those are the counties of Northern California that grow a lot of marijuana. And each plant needs 6 to 10 gallons of water a day. And they are growing, they're needing that water right in July when the river needs the water most. So some of these grows are drying up tributaries of the rivers and letting the rivers get stagnant and warmer because of the lack of flow. When that happens, if we have algae in the river, it will die, form these stressed floating mats, and the color here is yellow, but you probably could see some pink in there too. That's not a good sign. The temperature in these mats is getting high. Here's the water temperature that may be just 10 centimeters below, but in the water. And it's staying moderated by the water, staying around 20 degrees, which is comfortable swimming temperature. This would actually be uncomfortably warm for you to swim in. It's getting into the 30s, and this is what the temperature is in the sun-warmed, trapped waters on top of the mat. So what's happening to the algae? This is too hot up here for diatoms or for Glidophora, so they're dying. And now I want you to see if you can empathize with a diatom. Because here, remember, this is what the diatom looks like when it's healthy with all its carotenoids. But here's what it looks like when it's dying. It's lost those orange pigments. That green is unhealthy. And the green was its chlorophyll, which is now unmasked as the carotenoids have gone away. And they're bleeding their guts, their chlorophyll into the matrix. And all that nitrogen, phosphorus is going into this debris from cell death. But who's in that debris to pick it up but cyanobacteria that actually are perfectly fine at those warm temperatures? Many cyanobacteria like it hot, so we're going to have more cyanobacterial blooms in a warming world, and we've got to think about that. Here's the little knot here's a micrograph of it. It unfurls, grows, colonizes the good rusty growths of algae and blankets them with black carpets of cyanobacteria. And so here's a growing front, where the cyanobacteria are the black growths here, and the rusty red algae is here, and it's just spreading and taking it over. Well, if a dog jumps in the wrong backwater and gets this stuff in its fur and licks itself, as they do, they die, unfortunately, in 30 minutes, 20 or 30 minutes, in convulsions, because these particular algae in the cyanobacteria in the Eel River are neurotoxic. So that's not good. It's getting our neighbors' attention, and we need to figure out, as a community, as people who all care about this rivers, what to do about it. So we'll talk about that at the end, and it addresses your question about what we do about society, water, and aquatic habitats. But first, let's just sum up the food web, the stuff that we've learned. We're not looking at different organisms now, we're looking at the same organisms, but their numbers and their performances are changing as the environment changes. So here's what we've got after winter scour and adequate summer base flow. We've got the nice food web that feeds fish with edible algae. And if we didn't get that winter scour, we've got a food web where predator-resistant grazers take over, but the algae are still edible, and there aren't very many. But if we have severe summer drawdown, whether we had a big flood winter or a dry winter, big flood winters, it's almost worse because there's more algae to rot, you get overgrowths of the good algae by potentially toxic cyanobacteria. So this is really a worry. Here's the more general message. You have the same web members, but we all know that traits will work well under some environmental conditions and not so well under others, obviously. So drought, flood, there are many shifts in many environments that will change the strength of a path, a food chain or a trophic cascade through the same web, given context-dependent environmental changes in the performance of the web members. So that's a way of thinking about the first conclusion. Here specifically, we see that you need disturbance in rivers. You need that flood scour flushing to maintain fish, and you can also use flushing to clean up the water and flush things. The one good thing about rivers is they're really resilient in the sense that if you can clean up the sources of pollution to rivers, they're self-cleansing. Because of that unidirectional flow. Of course, then the sea might have to deal with what they're sending out. And what do we do to sustain them as healthy rivers? Speaking of the sea receiving it, the question about sea otters, I wanted to mention that some of the deaths of sea otters have recently been found to be due to blue-green algae that have been exported out of the Monterey rivers because they've looked at the livers of dead sea otters. Now, I'm not saying this is counting for those massive deaths, but they're finding over 20 dead sea otters because of river toxins from the agricultural rivers. So that's, and harmful algae are part of that picture is in that it partly is controlled by what we're doing to rivers and watersheds. So what we're doing locally is actually, despite the worry in the situation, this is some of the most fun I've ever had on the river because I'm meeting wonderful people who care about the river as deeply as I do and know it as deeply as I do, but in different ways. So we're sharing traditional knowledge and long-term residents and homesteaders' knowledge. And what these guys want to know is what algae do I need to thank because they're growing my fish. Actually, they don't need to know. They often don't. We always take algae for granted when it's doing its job, right? Because it's eaten and you don't see it very much. But it's really doing a lot of good in the world. And if you look at the microscope, they're very beautiful. But then they also do need to know which algae they need to be concerned about and keep their dogs out of those backwaters. So we're really having a lot of quite fun engagement and getting a lot of help from citizen scientists, tribes, and other partners in the basin. That's just great. That's what I mean by eyes on the world, but we call this eyes on the eel. It's great, great fun. And also, we've got a lot to learn. It's been extremely interesting to compare notes with tribes. Here's Ron Reed and Bill Tripp from the Kadu tribe, and they've visited our site. We've visited their sites a number of times. It's a good partnership because they have knowledge that's maybe 1,000 years old of how they manage forests to save flows into the late summer for the fish. And Californians are starting to listen and starting to learn, and they're also interested in the modern science that says maybe things have changed enough that we wouldn't do exactly the same thing now, but let's all see how we save rivers and forests and help our watersheds deal with all of these problems that we'll face under climate warming. So thanks, and I again welcome your questions. Yes.
Hi, I'm Samara from Bethesda Cherry Chase High School, and I was wondering when the Damascus, when they come in, even during the winter influx, do they still affect the environment heavily, as heavily as they do in the droughts?
I think, if I were to talk about their individual effects, yes, but there aren't as many individuals. So I think what a species does to a food web is partly how abundant it is, and then partly how strong each individual acts against or for something. And so you multiply those together. So it's the fact that there's just maybe 100-fold fewer that makes their effect less. It's a good question. Yeah. Yes.
Hi. I'm Maya from Montgomery Blair High School. And to add on to that question, since during the drought the caddisfly consume most of the algae, what do the tough midges do to survive, since in the food chain it didn't really show any other sources of energy for them?
They don't survive. Their numbers are way down. So it's not just the loss of food, it's the loss of cover. What they do is, it's kind of, you have to feel sorry for them. They get detritus. There's a little bit of silt, and they use their own silk to knit this stuff together. They make little jackets of silk, but they're very unprotective compared to the strong Cladophora tuft. So they're more exposed to predators and they're not eating as well. It's just lucky that they can rebound because mother midge can lay many thousands of eggs. So when they have a good year, their numbers are huge. When they have a bad year, there are a few females left, and a few are sufficient to repopulate it again if it's a good year. So that's how the midges make it. Yeah. Yes.
I'm Donna from Winston Churchill. So you said that some cyanobacteria are bad, but others are good. So what are ways that we could distinguish them two?
Well, it's a complicated question. I have to say I have awe, respect, and I would say love for cyanobacteria. They're just amazing organisms, and if there's a lot of great science to read about their evolution and what they did to the earth. So without the success of cyanobacteria in coming up with a very successful form of photosynthesis, we wouldn't have an oxygenated atmosphere. They're ancient. You've got to respect them because they're, I guess, 2.4 billion years old. They used to be thought to be 3.2, but some of the old fossils have fallen by the way in terms of people believing in them. But at least they're several billion years old. That's if you stand. If you look at the timeline on the HHMI ruler, maybe you can get a sense of perspective on that. And then many of that brown globular thing I showed you was also a cyanobacteria that isn't so toxic. So if they're in normal concentrations, they are doing an interesting thing for that river, which is they fix atmospheric nitrogen. Not all, but many do. And all the ones I showed you today do that. And that's good for the river because our river is very nitrogen limited, nitrogen hungry. If you add more nitrogen, that's part of the reason you're getting more production, is the activities of those cyanobacteria. So to make it even stranger, let me tell you that the favorite diatom that turns it rusty red has endosymbiotic cyanobacteria in it. There's a partnership. The shell has the diatom host, and then there are little deep green cyanobacteria. They're very ancient cyanobacteria. They've lost some of their functions because the diatom's taking care of those, but they're adding nitrogen to the diatom and making it more nutritious. So in that case, those cyano-endosymbionts aren't toxic. So it's very diverse and interesting. And in terms of good, bad, and ugly and all that, you know it's just kind of playful to say that. It's a way we can have fun with it and maybe remember it, but organisms are just doing their job trying to stay alive. And we have to figure out whether we need to fight them or learn how to coexist with them.
Thank you.
Yes.
My name's Maihan. I'm from Georgetown Day School. And I was wondering if you could explain how traditional methods help maintain the environment?
Yeah, I'd love to talk more about that. Thank you. What many tribes, and the Karuk are real leaders in this, say is that our great-grandparents, or our grandparents in the case of those guys, managed the forest often with cool fire right when the rain started. In the early wet season, they'd have cool burns. And in the forests of California, we get conifers that are Doug fir, and all of the trees except Doug fir can take fire. So the redwoods can take it, they stump sprout, and they have thick bark. So can the broadleaf trees, the madrones, the oaks, the bay. All of these trees do fine with fire, and sometimes they do better because fungi and epiphytes are burned off, and it leaves the trees healthier after a cool fire, the kind that they would use traditionally. But in the European land care management, we haven't allowed those cool fires. People get annoyed by the smoke, for example, or they are afraid of them. So the Doug fir then fill in, even a mature forest, they can grow slowly and fill it in. And then if we've done the clear cut and fire suppress policy that's happened throughout much of the West, you also get the same thing of a forest that's locked up. You can't walk through it, you can't see through it, you can't look under your knee and say, yep, no deer up that hill, which is what they wanted to do. But also, then you get the mega fires, and the trees aren't as healthy, they don't grow as well. And there's a huge leaf area to suck water out of the rivers and evapotranspire it into the sky. So that dries up the streams. That's, in addition to marijuana being one of the culprits, it's the dugfurification of the eel basin and similar basins that is part of the land use that we may need to rethink to deal with drought. Yes.
Hi, I'm Becca from Dominion High School. So the harmful algal blooms that you mentioned can be toxic and are known or highly suspected to affect certain organisms. There was recently an experiment on how they affect spatial navigation in California sea lions. So is it possible that, like, do you know if this toxic algae could potentially affect bird flight? Because if they can't navigate, then how will they be able to migrate or navigate in the air?
I don't know about that. I know some flamingos eat crustacea, but some eat algae, and I think they have adaptations, I believe, to deal with cyanobacteria. So there are actually some fish that can eat cyanobacteria too, including some that are very liver toxic. African fish can eat it. So some vertebrates are either resistant or have enzymes that can break down the toxins, and I don't know about birds in general, whether they'd be affected. That'd be an interesting area for research, especially with birds that feed from the sea a lot, where the poisons might be concentrated in their prey, albatross and stuff. Yeah.
Thank you.
Yeah. Yes.
Hi, I'm Megan. I'm from National Cathedral School. So I was wondering what's currently being done in the realm of environmental policy, specifically by the government of California to deal with the diversion of water. Just because I thought the point about the marijuana was very interesting and how the diversion of water culminates in this phenomenon. So is there anything being done?
Yes, there is a lot of management at the state and mostly the state level and some county level. And so it's not being managed yet, but they're thinking about it. First, they're just trying to get a handle on how much water is diverted. And then it's a little bit worrisome because this is a kind of a frontier mentality where both the tribes and the European Americans, which call themselves settlers, the ones that are really kind of homesteading, they're very independent-minded, and they don't want people looking over their shoulders, as you can imagine. So it's kind of the Wild West in some ways, and they're going to have to change that. But on the other hand, there's a lot of goodwill and neighborhood spirit. Everybody wants the salmon to continue. And so there's movement in the state to figure out how to account for and watch water use and water disappearing. You can see the marijuana grows from Google Earth now, and some fish and wildlife agents who are very heroic have jumped out of helicopters to see if we see this damage from Google Earth and these many greenhouses, how many plants are there? So they've done all that. But the other thing that's working, I think, there are two reasons to hope, and one is that groups are persuading neighbors to store their water in the winter not in dams, not in holding ponds, because those collapse and send more fine sediments into the river, and that's been a problem, but in fiberglass or plexiglass tanks, huge tanks. So if you store the winter water and then meter it out in the summer, that will be a lot less damaging. Then the other thing is we're hoping that legalization will make marijuana a lot cheaper so that the motive to grow it in the really damaging ways and the hard to get to more wilderness areas will subside. Cross fingers. Yeah.
Welcome to HHMI's 2016 Holiday Lectures on Science. This year's lectures, Ecology of Rivers and Coasts, Food Webs and Human Impacts, will be given by Dr. Mary Power of the University of California, Berkeley, and Dr. Brian Silliman of Duke University. The fourth lecture is titled Climate Stress and Coastal Food Webs. And now, Dr. Brian Silliman.
Thank you very much. I'm excited to be here in the second lecture to talk to you about solving mysteries of ecosystem decline. And in this talk, I'm going to use the ecological and scientific methods, science, experimental science, observational studies, and collaboration to try to solve the decline in coastal wetlands that's been occurring over the last 20 or 30 years. And unfortunately, in marine ecosystems around the world, it's a story that's too common. There's been drastic declines, for example, in oyster reef habitats. Over 90% of the oyster reefs around the world have declined. We've lost those. Coastal wetlands, including salt marshes and mangroves, it's about 40% decline. And for coral reef, it's 50 to 60%. So somebody asked a question about what can scientists do, what can the public do. One of the things scientists have to do is they have to step out of understanding just conceptual ecology and jump into those arenas and help solve those problems. What types of interactions are leading to those declines? And if we change the strength of those interactions, can we help these ecosystems recover in these systems? And a lot of times what's going on at the same time we're losing marine ecosystems is that climate change is leading to increased physical stressors. Temperatures are warmer, the oceans are becoming more acidic, and drought, high intensity of droughts, they're becoming more frequent. So we have more frequent, intense droughts. At the same time, we're altering food webs. We're overhunting, we're overfishing in certain areas. It changes the number of predators and grazers. And there's typically two camps of scientists looking at the declines in these ecosystems. One that says, oh, it's climate stress, so physical factors. Another one says, oh, it's the change in the food web that's showing the decline. And there's not much looking at how these two factors interact. Could climate and food webs be interacting to either protect or stimulate synergistically the decline in these ecosystems? And today I'm going to look at whether or not we have interactions in coastal salt marshes. And over the past 15 years, we've had 4 100-year droughts. These droughts last 2 or 3 years. They increase salinities in salt marshes, spiking the salts concentration in the salinities 30 to 60%. And that's 350,000 acres of marsh. That's over 10% of the marshes we have in the United States have experienced dieback in just 15 years. So you can imagine people are pretty overwhelmed and upset. State of emergency, the governor of Louisiana started a state of emergency about the die-off of these salt marshes. Bringing in scientists, we have to solve this mystery, figure out the environmental degradation that's going on, and try to reverse that. In salt marshes, the prevailing theory is that bottom-up forces or physical factors are the ones you need to worry about. They control the plant growth. The grazers really aren't important in these systems. So what is the physical agent that's changing? And everybody saw this drought and pointed to that. It's the drought stress that's killing these plants. And here's an example of a big area that's been killed off in Louisiana, in typical areas the size of football fields, double that size. And we have something that's very unique. We have a dying and decaying ecosystem juxtaposed to a very healthy one. And they seem to be expanding over time. A lot of theories came about. They spouted up about what was causing the marsh ecosystem die-off. I was following it, thinking about it, and I got a call from a reporter who was going to put forward a theory based on my work. And he did in about 2 or 3 weeks. He put out there in the newspapers, because blue crabs were declining in these areas at the same time the salt marshes were dying off, he proposed an alternative hypothesis that what I had said in my studies, that blue crabs, by controlling snail densities, indirectly facilitate plants, could be happening at large spatial scales. The loss of blue crabs could be leading to outbreaks of snails and die-off of these marshes. Most of the scientists, almost all the scientists in the Southeast said there's absolutely no way that would happen. We've looked in our marshes and we see dying and decaying plants, and we don't see snails in those environments. So I got in the truck, and I wanted to go check out all these areas. And I called up people who didn't believe me. I said, take me to your die-off areas. Let's check them out. Let's see if snails are interacting in these environments. And this is a typical salt marsh die-off that we've come upon. With the Georgia, Louisiana, North Carolina, South Carolina. Again, these large football, soccer field areas, dying, decaying grass with patches that are left, some doing better than others. And then this very distinctive line, this demarcation between a dying, matted down ecosystem and one that's starkly opposed to that is pretty healthy. And so if you get out of your truck and you just don't stop there and you start to look at the environment and look for potential strength of interactions. This is what we found as we walked across that, is that we went to this line and we found that there were super concentrations of snails right on that line. And if we walk down that line between the healthy grass and the dead grass, it's about 2 meters wide, and the length of it was between 200 and 500 meters, of 2,000 snails per meter squared in those areas. That's millions of snails, and we term that a consumer front. Super concentration of grazers in one area is making a front like an army, and it looked like it could be plowing through in those areas. And we went to Louisiana, Georgia, and we took our quadrats out. We want to count the number of snails. So you put it down, and it would take almost an hour in each one of these plots. And one of the scientists who didn't believe me at Louisiana State University, I said, take me to your die-off area. We got off the boat, and he said, no snails, none whatsoever. I said, let's go over to that other area where it looks like there's a decaying band. Let's see if grazers are there. And lo and behold, we walked over the marsh mud flat. Took us a long time. We got stuck in the mud. Got over there and saw lots of snails. And he said, I believe you. Let's get to work. So we did the quadrats, and then we pulled the snails off, and we tried to look for snail grazing effects. And here's an example, another picture. The plants that were in this die-off area, the ones that were left over, were looked to be overgrazed by snails as well. And it looked like the snails were killing them. And this is a big effect for a snail to have, to kill a rooted vegetation. But we need to test that experimentally. So based on these observations, we proposed 3 different questions. Do snail fronts generate marsh dieback? Do they contribute to the loss of this ecosystem by overgrazing? Is this front pushing the die-off of the ecosystem further in the healthy systems? What factors contribute? Why are these snails piling up? Something counterintuitive humans don't necessarily do. We're a little bit claustrophobic. But all these snails are concentrated. It looks like a powerful edge stress that's out of control in these ecosystems. So what factors could do that? And can snails and drought interact to cause marsh dry-off? This is looking at climate stress and food webs. Is there an interaction there, or are they not doing anything to each other? Let's use experiments and modeling to test these ideas. First, using one of my favorite tools, the cage. We go along these snail fronts here, and you set up a control area. So this is a control snails, snail removal, and you come back over time. And this is what we found time and time again at all these die-off areas. We saw that snail fronts were moving through, and in the wake of the snail front, you found an exposed mud flat. They were killing the vegetation. How is the marsh grass doing inside those cages? Well, if you look over the top of the cage, it's doing just fine. And these are the plants that are exposed to the drought stress. It's supposed to be killing off and driving death in this ecosystem. Clearly, physical factors are not killing the marsh in this particular instance. In fact, the drought had subsided, and those physical stresses were now gone. So this experimentally demonstrates that snails, a top-down force, is driving die-off in this ecosystem. Ecologically, it's pretty amazing because these plants have small potato-like storage units called rhizomes, and they have resources to regrow. A lot of times, grazers go to greener pastures. These snails are sticking on these plants, likely because snails are really after the fungus. They facilitate in the plant leaves, and as they graze down on those plants, they become better substrates for fungus. There's no reason to move off those marshes. So what we found in all these different sites that the rate of die-off was increasing at different rates. So does the number of snails that you would find in these snail fronts really to determine how much ecosystem you're going to lose? And we found a strong correlation there. That with increasing snail density, we saw increasing loss of marsh after 3 or 4 months. And this is in Louisiana, Georgia, and a variety of other places. We also found an interesting, what we thought is it looks like an inflection point. And you see this right here. Below this line is gain, salt marsh gain, so a little bit of recovery in the marsh after drought, and this is loss, continued die-off of the ecosystem. And about right here, it looks like there's a threshold. And that's a point in an ecosystem. As you're increasing stress, in this instance, it would be stress from grazers. As you increase that stress, is there a point where that plant or animal can no longer take that pressure and then it collapses? Okay, it disappears spatially from that spot. And we seem to have found this. It may take 1,000, 1,200 snails per meter squared for this process to generate. So what factors, okay, could change in that ecosystem to lead to a piling of snails at that density? Could it be blue crabs? The decline in blue crabs, they were falling at the same time this was going on. Both drought issues, they don't like drought, and also there were some overfishing suggestions that were going on. But let's look at that relationship. And you've seen this graph before. It's a relationship between crab abundance and snail density. Before you just looked at the relationship, but pay attention to the numbers, the quantitative numbers. In years where we have low blue crab abundance, the snail density does go up, but does it reach that threshold? It doesn't per meter squared in areas where die-off's not going on. The snail numbers double, triple, but they don't go up above 1,000 per meter squared in those areas. So it suggests that the decline in blue crabs could contribute to increasing snail densities, but not necessarily get them to above 1,000 per meter squared in the areas. Could they be interacting with drought stress in the environment? So to do this, we go to experiments. We go to healthy marsh areas that haven't experienced die-off, and we manipulate the amount of snails and the amount of drought stress. And the drought stress is pretty easy to manipulate. You take 50 milliliter centrifuge tubes, you take a drill and you drill holes in the side, you take pantyhose and you wrap up salt, stick that in there, and you have a salt shaker, underground salt shaker. And you stick that in the ground, and you can elevate the salt in that muddy environment to levels that mimic what happens in drought. So we have a drought stress, and then the snail stress is easy enough to manipulate. So this is a picture of a healthy ecosystem right here, of one without drought and without snails. And I'll show you the striking pictorial results there. This is a snail exclusion, a very healthy marsh. And if you add salt without snails, you get a strong negative effect. 50% reduction in plant biomass when you increase the amount of salt stress in those ecosystems. So it suggests that drought is having a strong but non-lethal effect on these ecosystems. So this experiment, predicts that drought alone is not doing it. What happens when snails are there? And you have a marsh on snails, where you have a controlled treatment. Again, you can see top-down effects of snails that reduces it 50%. Adding 50% reduction plus 50% reduction is easy math, and it leads to 95 to 100% reduction in biomass. And this shows when you simulate both structures in the environment, if you increase drought, top-down control is increasing in that environment, and you add those together, and you can experimentally generate die-off in the system. And it shows those climate stressors can be interaction with top-down control, could be driving die-off in these systems. Well, we know from our natural history of the salt marshes that when drought occurs or lack of water coming into the system, salt doesn't increase equally in all parts of the marsh. It does so more in the high marsh. And so that means that this is going on, we're getting pockets of die-off. We're getting mudflat. But what do the snails do when their prey item's gone? You can only guess. They're probably going to go after the prey item. That's what we thought too, but we wanted to test that. That is the plant. So here's what we did. We went to the mudflat where some of these die-offs would occur, and we took the snails and we marked them with little paint. And then we spread them out. And we asked them, which direction do you prefer to go if grass is 2 meters away and the mudflat's on the other side 2 meters away? They have decent eyes, and they can tell visual acuity. They can see if something is off in the distance with height, for instance, with shadows. So we put 200 snails out there. Amazingly, after 2 hours, this is amazing for snail movement. I'm used to working with snails. They don't move more than a meter horizontally in a salt marsh over a year period. They just stay going up and down those steps. They don't move much at all. 2 meters in just about 2 hours. I was amazed. And then 96%, everybody was moving towards the marsh. And then we went back further, and you could do it 4 meters and 6 meters, and they stopped being able to detect the marsh about 10 meters away. So they're going towards the grass, and they're going fast when they don't have plants. So it's a behavior in which they home to their prey item. And look at this movement, how it changes. 16 meters per hour. It's very fast for snail on mud. And then when they get here, it's 25 centimeters per hour, but it's back and forth. They just stay in one spot once they get the grass. So this is a visual representation of what happened. You take all those snails, 96%, and they move over to the edge, and then they stop. And because of that behavior and because of the spatial heterogeneity in the system where you have no grasses and grasses, it then creates a wall. It's like taking the band during a college halftime show, and then everybody moves to the sidelines, and you have a super concentration of individuals right there in a big band. And right there, you go from a situation where you don't have high enough densities to overwhelm the system to one where you're likely to go eclipse a threshold that we've identified before. So if we look at snail front movement, if we hit that threshold, are we going to see movement over time? And let's follow that a little bit more than just with a cage. So these are in Georgia salt marshes. This is the amount of marsh grass as well as snail density on this axis. And then snail density over a distance is denoted here with this orange line, and then marsh grass with green. 0 meters demarks the differentiation between a dead ecosystem and a live one. So over here we have green grass, and over here we don't have grass and we don't have snails. And we can see over time, this is time 0, 3 months later, the snail front's moving across 5 to 10 meters, and in its path, it's killing off the grasses. So it's expanding. Remember, this is happening over a large circle. 400 meters long. So you have to multiply that times these distances to get an idea of how much of the ecosystem they're killing off. Then we get one more time sequence. The front is actually maintaining itself and continuing to go through the system. A little bit scary situation. Importantly, during this period, the drought had subsided. So it looks like we set up a potential catalytic event where you have a wave that's propagating through an ecosystem, despite the fact the original stress is gone that initiated that concentration of grazers in the system. Potentially dangerous. So we're interested in what potentially can control those snail fronts. So if we put our die-off sequence together, it looks like we have climate stress and top-down control interacting to cause localized die-off. We know that from natural history and experiments about where that, and then once that happens, very importantly, we have the behavior of the snail comes into play. And because they home into those plants, they all leave the mudflat and go to the sidelines, and then you get this edge stress going on. If they were still spread out, you wouldn't have the threshold eclipse. That eclipses, you get over 1,000, 2,000 snails per meter squared on the sidelines, then you've hit the threshold, and then you get this runaway or catalytic effect that despite the fact that drought has subsided, it continues to mow down this ecosystem. Very powerful phenomena, interaction between spatial processes and food webs that we haven't really thought about a lot in ecology in these systems. So really big effect. If we can model this, this is fun. You take your math classes, you think it's where you're going to apply the math. It's amazing how much you use math and science. And you can use a lot of calculus when it comes to looking at the functional relationship between cause and effect and X and Y. You can do that in modeling. And this is a simplification of the model. But we wanted to know, can we actually generate this with math? Can we generate what's going on? And so this simulates, this is distance right here. This point right here is where we have a mudflat, a die-off. In this area over here, these are snails. So that's snail density in the healthy area. Here's a snail front. And this is the amount of grass in the area. And so we put in there information about how the snails move. They don't do it randomly. They go towards the plants. So the snails in this front, guess what? Cannot go backwards in the model. They have to go forward. And as they're going forward, imagine the chaos that's ensuing as you have snails here in the healthy environment, and there's a front of snails rolling over. And they're going to be incorporated in there. Remember, they don't move too fast if the mud's not going. So this should expand over time, leading to faster degradation of marshes. And indeed, when we run the model, that's what we find. It's what we find in nature. It's not perfect simulation, but we can do that. We can apply this model to other situations. And we can change things. How important is that behavior? Well, we take the homing out of the model, and the fronts never form. So it's just a localized effect, and you don't get the domino effect. So that behavior becomes really important if we potentially see this in other places. So one of the things people usually ask about is, it's just going to kill all the salt marshes. Is this going to stop? One of the things that happens is that the winter comes, and these snails will migrate when it gets cold, and they'll leave the fronts, and they go up to the high marsh near the terrestrial border. So the winter is, the coldness will stop this in action. And there's also a mechanism for recovery that moves faster than the snail fronts in many instances. And that is, we just published this paper, and I think it's very exciting results, and we use models and experiments to get in the field. And so what we did, we measured the recovery rate. How fast do these patches of mud fill in with plants? We did some experiments. We sprinkled seeds out there, and seeds cannot grow. It's so anoxic. Without oxygen, they all die. And so the only way these plants can grow back there is vegetatively. The roots go under. It's like a weed, crabgrass going in your driveway. They send these runners. They're feeling, and they poke up through the surface, and then they establish themselves. And they can go 1 meter or 2 meters a year. And the rate of coverage is based on how much perimeter of the marsh is facing that area. And what we found is that if you don't have any of these patches in here and you just have a mudflat with a circle, it takes a long time for those places years to recovery, 20 to 100 years depending on the size of the mudflat. But if you have these little patches that are surviving, and we found out that those patches survive if they have muscles. The reason they're survive is there's a muscle mound underneath of them. All these rib muscles that create what I said earlier was a spa in the marsh. They hold water so drought doesn't increase salinity there. There are predator crabs that will actually eat the snails, and it's like a tree fertilizer stake. You put it in there, and they're pooping right underneath the Spartina plants, the marsh plants, and it increases. So these plants that are living on top of mussels are very robust, and they can withstand a snail front. They survive, it goes through, and then we get regrowth. It's a nuclei for regrowth, resistance. And look how fast regrowth occurs, 10 to 12 years. In some places, it can even 5 years. So this mutualism underlies the ability of these ecosystems, marsh ecosystems that you have right here on your coast, to not disappear. It's those mussels that allow it to deal with those consumer fronts. Pretty cool interaction. So we're talking about climate stress and food webs. Let's put them together. We have a new marsh food web that incorporates bottom-up factors influencing the plant growth, conceptual model here, top-down control by snails and blue crabs, indirect facilitation. What is climate stress? What is climate stress doing in these interactions? We know from experiments right here, this is getting stronger, so it's amplifying that. For blue crabs, what drought does is that blue crabs really prefer waters that are about half salinity of the ocean. Turns out in full ocean salinity waters, they're very susceptible to disease, and they can move. Before I told you that they could swim, and they move up rivers. As the ocean moves in drought, because not as much fresh water is coming down the rivers, they move with it. And so they move off the marshes, and we have less predation in those years. So what happens is we have drought stress that's affecting the interaction strengths here. It's amplifying top-down control by grazers and cutting off the top of the food web effectively, removing this facilitation cascade. So that we see a strengthening or triggering of outbreaks of grazers in the system. Two additive effects. Very powerful, and it's leading to less of an ecosystem that a lot of people enjoy and depend on. Is this happening in other salt marsh systems? Asking generality is an important part of ecology. I've been working with collaborators in China, and the same thing is happening. They're having increased frequency of intense droughts, and they're losing one of their prized natural resources, salt marshes that are uninvaded by plants from North America. They have these beautiful native salt marshes that turn maroon in the fall, but they're all dying off, and it's transforming into a lunar landscape. So they're going after drought. Everybody's pointing the figure. It's just drought, salt stress, and after drought stress, they go out there and plant plants, except all of those are dying even when drought stress is gone. When we go to if we go to different river basin, we see the same thing going on. Is there a possibility that grazers that we've seen in our last survey that are in these marshes are playing a role in this system? So we started studying about 4 or 5 years ago, and we put cages out to look at before drought in the anticipation that we would get another one to see the effect of drought on a system with and without top-down control in it, the environment. So we used cages again, and this is 2002. 2010, this is pre-drought years. And in 2011, there was a big intense drought. And this is what happened in control cages and exclosures. In control cages, we got nearly a complete die-off. A lunar landscape emerged in those environments. In the caged areas, we saw that there was drought stress. It suppressed the plant's growth, but it didn't kill them. The ecosystem was still intact. When the drought subsides, you can predict what happens here. You get recovery where you do have plants, and you get continued decline here in those areas. And what we found is that crabs formed fronts just like the snails, and they were more mobile. So when all the plants were done, they would move around and go finish things off, and anything tried to regrow into the area, even on a mussel mound or anything else, they would clip it down. So having big impacts in those systems. And they suppressed it from even recovering in those environments. So that suggests that it's a common phenomenon in salt marshes where climate stress is going to amplify top-down control and lead to loss of ecosystems that we really care about. Is this something that we should worry about in other ecosystems that we care about? Forests, rivers, coral reefs? Or is it something that we can just put to the side and say it's only in salt marshes, and if you care about salt marshes, you should care about this interaction? Well, let's go to ecological theory. In ecological theory, we have a very simplistic model that does predict. It's called the environmental stress model, and it's based on work in rocky intertidal systems, where they studied sea stars eating mussels. And what they found was that the stress on the foundation species by the consumer, in this case the predator, the sea star, decreased predictably as you turned up physical stress. You go up in desiccation stress or wave stress. The sea stars couldn't hold on as well as the mussels, and they couldn't withstand as much desiccation. So they predicted in most situations, as you increase physical stress, consumer pressure would go down. That's not what we're seeing, but theory suggests that should be a common phenomenon. If that's the case, as physical stress is going up, there's an obvious answer to the amount of stress on the foundation species from physical stress. It just goes up as well. And then you have crisscrossing inverse lines here. If you're measuring total stress and it's all about grazers or climate, they kind of equal each other out in this conceptual model. Okay, it's imperfection, it's not numbers, just an idea. And we have total stress in the system that's flat. You trade one for the other. So is this happening in other systems? And if it is, in our models for understanding climate stress, we don't have to worry about grazers and food webs. We can just turn it off. Just try to worry about acidification, temperature. But if that's not the case, then we need to bring in food webs too. So one of the other examples I was reading in USA Today probably 10 or 12 years ago, looking at this massive loss of forest out on the west coast of the United States. And it was driven by drought stress and interactions with grazers, these pine bark beetles. They carry fungus with them, and they drill holes in the sides of these trees, and the fungus they carry with them infects those trees. On non-drought years, the plant's immune system will form a wall around that area and keep the fungus from moving into the plant. On water-deprived years, the plants don't have those resources, and the fungus, hyphae, proliferate in that plant, and they kill it. They can kill entire trees like this. And those can have massive consumer cascading effects. You can get big fronts moving that you can see from a helicopter, not when you're hiking, and they move through these forests, killing a lot of these ecosystems. So this suggests that a lot of these are going on. Here's some numbers behind that. If we increase the intensity of drought and frequency, are we going to see more of this? Here's for snail fronts and for pine bark beetles. What we find is that this is the Palmer, these are drought indices. This is very dry, very wet, very dry, very wet. In the area of beetle kill and the number of snail fronts. And there's exponential relationships in both of those. As you get to this, looks like a threshold here of a -2 or -3 drought, and the number of snail fronts escalates. We start to have this process that we now know about because of models and experiments is happening on large spatial scale. And the same thing, it looks like pine bark beetles. We get, look at that, how much area is being killed by pine bark beetles is proportional to the amount of drought in the area. So how common is this? We have two examples now. Let's comb the literature, go to the computers, look at the journals. Are people seeing these interactions? And what about these consumer fronts? These are really powerful edge stresses that can devour whole ecosystems. Are these common in other situations? So we looked at grazer outbacks and consumer fronts. What we found is there was actually 16 examples. That doesn't seem like a big number, but that's 16 different ecosystems in which we found consumer fronts can kill the entire ecosystem. It's not just happening on a small scale. I knew about urchins going into it. And this happens throughout the world. This happens off the coast of, it's in the North Atlantic, the North Pacific, the South Pacific, and other areas. It's a very common, and they plow over the kelp beds. Locusts can do this in agricultural systems. And this is the one I teach in coral reefs, but I wasn't even thinking about this. It's not really called that in the literature. But the disease that's driving a lot of the coral reef die-off in these ecosystems around the world are banned diseases. And they're microbial communities that form consumer fronts. And in the back of them, they leave an exposed calcium carbonate skeleton that's colonized by algae. And in front of them is a healthy coral that forms the foundation of those ecosystems. So here's another consumer front. What's potentially setting that off? In 10 of the 16 examples, physical stress. Okay, 10 out of 16 examples, physical stress was stimulating these consumers to congregate and overwhelm their prey item, and then lead a threshold where they could continue that progress without the stress and present. And importantly, just like the snails, all of those animals had this characteristic. They always moved towards their prey, resource-dependent movement, and they didn't mind being in a closet with other individuals in the same species. They weren't claustrophobic, okay? And so if you put them all together, they don't move faster away from each other. They just kind of slow down. So the urchins do that, microbes do that, and locusts do that. Here's some more examples. Wildebeest will do this. It's driven by rain deprivation, gypsy moths, and also crown-of-thorn sea stars. This is, no kidding, it's the thorn in the side of coral reef managers throughout the world. These crown-of-thorn sea stars are outbreaking, and you've all heard the story. You cut them up and divide them up, and they reproduce, and they come back. They're killing entire ecosystems. Hundreds, even thousands of kilometers squared of coral reef are dying in those areas. So it seems to be a common phenomenon. What about biotic attack on drought? Drought is a problem. It kills agricultural systems. It kills plant systems that we depend on for resources. And people are studying, they're trying to predict at what level of drought, how much drought can we withstand before we lose the ecosystems we care about? We're trying to predict that. And all those models, none of them include top-down forcing in the system. But if you look at the studies where people are noting the natural history, some observations about their studies, almost 80% of them note that there's a biotic attack going on. Different players, remember that? We talk about different players, but the same process. These plants that are under stress are being attacked by pathogens. Nobody's done the experiments to test their relative importance like we've done in salt marshes. So we don't know if they're contributing in a big way, but it could be really, really important. It suggests it's a little bit of a smoking gun. So let's revisit theory. So this is the simple environmental stress model. And let's reevaluate this. So at 10 of 16 examples, physical stress was actually increasing consumer pressure. That's the opposite of what that's telling us here. Remember, consumer pressures in this prediction should go down as physical stress goes up. And so if you're over here in a very climate-stressed ecosystem, that's what you have to worry about, is dealing with climate stress. But the opposite could be true, and that is consumer pressure could either be flat, or we have in snails, it could act synergistically. And then look what happens to total stress in the ecosystem. It goes up, and this is the stress on the foundation species. And somewhere along this line, we're going to put a red arrow. And that's how much, that's the threshold of that ecosystem. That's how much it can withstand before you start to see decline. And we need to find that. We need to find where that's at. So this helps inform how we compose our resistance and our resilience models and recovery models in ecosystems. And let me give you an example. This is understanding ecosystem resistance, these thresholds. It's in all the papers, in science papers, in the New York Times. People, what's the point? What's the tipping point? How much can these ecosystems handle? Because we'll manage it up to that. We'll find that point, and that'll help us, when we have limiting resources in conservation, allocate to the ecosystems that are about ready to go. Find that threshold. Well, okay, here's a relationship between physical stress and the amount of a foundation species. Marsh grasses, mangroves, coral reefs in those areas. As you turn up climate stress, you can have a relationship that's just linear. So you just turn it up 1 degree, you get a proportional decrease in foundation species of 1%. It could be a 1-to-1 ratio. That happens a lot of times. You saw it with snails and marsh biomass. What we're finding in a lot of ecosystems is they have some internal feedbacks that allow them to withstand a little stress, like warm-bloated organisms. They're able to deal with some of those stresses within physiological adaptations. And so you don't see a change in the amount of that species for a while until it's eclipsed. So we see a flat line as we turn up physical stress. But it gets to a point it's just too much. You have too many exams on one day, you just can't handle it, right? And then boom, you get to that point. We all know about thresholds. That happens in your 5th exam. You're like, I just got to get out of here. I can't even deal with that 5th exam. And you get this threshold. And that's what we're searching for. So we do experiments with different levels of salt stress, different levels of grease, and we try to find those in ecosystems. We have one in salt marshes. But all these models make a basic assumption, or they just forget to include it. As physical stress is going up, they really don't include in these models, quantitative models, what could be happening to the grazers and predators in the system. They're not incorporating that. So we're managing for that. In which way should that threshold go if we have a system we have additive effects with consumers? Right, to the left. And this is what's likely going to happen. So we're going to have a lower threshold. It can handle less drought, some of these systems, less warming in these systems than we think, or less, for instance, less acidification. If the consumers, the grazers are going to say, here's a prey item that's weakened, I'm going to attack it in those environments. So that's important to guard against. So it's a bit overwhelming. You're like, well, this is a little bit sad to hear all this. They're more vulnerable than we thought. But this is actually empowering, because it tells us in many of these systems, if they are more susceptible to graze or outbreaks, as physical stress increases, we need even more predators around. So we actually have an answer to that, to push their threshold back up. These grazers right here, if we got rid of those, either through harvesting them or actually having more predators in the system, we may be able to increase the threshold back to where it naturally is with physical. So here are some key findings. We've talked about trophic cascades, but now we know by looking, having an open mind, and looking at interactions between physical and biological forces, that climate change, just like loss of predators, can also unleash grazers on an ecosystem. I'm calling that a climate cascade in those systems. And it may be happening in a lot more ecosystems than we imagine right now. And so they can also act together. So if the predators are being lost from these systems, you have less sea otters, or you have less blue crabs. At the same time, climate's going up, it's going to be even worse, and the threshold's going to come down. It's going to be synergistic, not just additive in those systems. And once you know something, you can take it to a new system. So this puts the alert on. We just didn't stay with salt marshes. You go look for generality. You look at other ecosystems. You talk to other managers. Is this something you're seeing with urchins? And we start to do generality, and that will help in the conservation of these systems. Is the climate for threshold stress is lower, but we need, how do we combat that? We need more otters. We need more blue crabs. In those times of drought, we need more predators in the system. Or, for instance, if the otters aren't there, we might increase harvesting of urchins for the fishery during those years, because fishery management could compensate for this. And importantly, I think it's, you read a lot about theory, about how systems are supposed to interact, and you've seen that in the lectures here today, that observations that you make yourself in nature may not jive with that. And it's important to trust your instinct and say, okay, let's evaluate that hypothesis, use experiments, work with other scientists, test those ideas, and confront theory with data. Theory is fantastic because it sets a conceptual idea and a model for your understanding of how that system works. And you find something that maybe doesn't jive, you test that, theory isn't correct, and then you incorporate it in there. So we've expanded. We haven't rejected the old bottom-up paradigm in these systems or physical forcing only. It's both. It's not one versus the other. It's both top-down and bottom-up control of these systems. Thank you very much. Goes anybody in the top up here? Maybe in the middle?
Oh, hi, I'm Erin from Episcopal High School, and I have a question. Are there any successful cases where you introduce like a predator and you help to push the threshold to the right?
So that's a great question, because as these predators, we have conservation for top predators that were overhunted. They're coming back, and so they're moving into areas. Wolves, the reintroduction, there's a little debate about this, but the idea is it's changing elk populations and it's allowing forests to regenerate. So that was an introduction to save the wolves, but there are consequences on that. The sea otters is a good example in salt marshes on the West Coast. The sea otters have been undergoing conservation. Their populations are increasing, and they've moved from what we thought was their favorite habitat, kelp beds, very agreeably moving into seagrasses and shallow environments, and they seem to be growing well. So we may be wrong about what they prefer. They're in salt marshes now, and they're eating the crabs that have been killing the marshes there, and we're seeing a reversal. We're seeing that edge loss that we saw before on the marsh edge where all the crabs are burrowing, crab condominiums, is not eroding anymore because there's not any crabs there. And the plants are rebounding as the otters come in. So top predators can rescue these systems in these examples. And we need to know how much more often this could be the case. Great question. Top predator rescue. I like that. Back here.
I'm Larry from the New School of Northern Virginia, and I'm wondering if adding mussels to salt marshes could like reinstate the natural state of the marshes.
Right. Love that question. The answer is yes. Here's another paradigm we need to think about. Restoration of marshes and protecting. We've taken all the theory from pine forests, where you plant everything away from each other, because competition is supposed to be bad. You don't want to be near a neighbor. It's the complete opposite in salt marshes and wetlands. The closer you plant plants, they help each other by shunting oxygen down there, putting mussels in there. They don't compete for space like they do on the rocky shore. They actually increase plant growth. So we're doing that right now, but it's the scientists working with the conservation organizations who aren't reading our papers. And we're saying if you just walk a little bit further and get the mussels over here and plant them next, it doubles and triples the rate of growth in those areas. And so the generality of that is really important. So these ecosystems are declining, and conservation typically, and the paradigm is that we have to identify a threat and reduce that threat. So get rid of negative interactions, okay? That's really important. The other thing you can do is you can enhance positive interactions, just like in top predator rescue, another mutualism like that. And so even if you don't have threat reduction, and you increase positive interactions like this, you can overwhelm that stress and shift your threshold out even more. So that's, that right there is a sort of shining light in all this, finding these species interactions is we gotta, and we have to do that systematically. When I sit at conservation tables, everybody's like, what are the threats? Let's get rid of them. What are the threats? Let's get rid of them. And now I say, what are the positive interactions? Let's get more of them. And not do it randomly, not just predators, but mussels in those environments, microorganisms, everything. Let's think of every positive interaction that could occur. Great question. In the middle up there, the top. Mm-hmm.
Hi, I'm Emily. I'm from Oakton High School, and I did a lot of research in the past year on the crown of thorns starfish and how it's affecting the Great Barrier Reef. And my question was, there's a recent development that scientists have made in Australia where they are using the stomach acid of cows to kill the starfish because they're allergic to it. And my question would be, if there's a situation like with the snails and the crabs, where the crabs do not naturally stay stay near the snails in a stressful environment. Would you condone like a path leading to the like death of the snails? Would that be a logical like way to go, or would you like want to go a different way first?
It seems like a logical way to go if the method was something similar to like you described. That's really, I didn't know about that. That's really fascinating. And that the consequences in that method are just the killing of consumers in that front. And there's not consequences on the rest of the ecosystem. And a lot of times we've gotten that wrong by introducing other species that we think that will just kill that one species and they start killing everything in the ecosystem. So we have to be very careful about that. But yes, we've talked about with urchins, when they have urchin fronts, they've actually talked to fishermen and they open the urchin fishery so they'll take more of those. So managing those consumer fronts to kill that many snails is difficult because you'd have to spray a molluscicide, and that is not something that's nice to everybody. It kills oyster reefs, it kills the mussels, it kills all kinds of stuff. So we have to go to snail picking. Maybe for up here in the top still. I'll come down here. Right here. Sorry.
So are predation relationships in resembling like feedback loops, are they necessarily like a determinate correspondence phenomena, or are they somewhat dynamical chance encounters? And how would that impact the reliability of quantitative models in predicting future events?
Oh, great question. So the predators finding these prey, like blue crabs finding the snails,
right? Are they?
Is that random, or is it they actually more
like
likely to find them in these clumps?
Like, is it indeterminate? Like, also considering the spatial heterogeneity among.
So the blue crabs, if they move into the marsh, and they move in there pretty fast, and then in the areas without plants, they actually move faster and they go towards plants. So there's a little bit of determinism there. And then when they get to the snails, they stop on that patch. And they sit on that patch until they can't feed anymore or another predator shows up. So there are some feedback. As the mudflats appear, the predators will actually find the prey to a greater degree. The problem is there's not as many blue crabs in the environment. So you don't get this feedback where as snails start to concentrate, predators compensate and they find them and sit on that. One of the interesting things we're finding that may be a compensatory feedback is that these snails get infected with parasites. And the parasites stop them from climbing because the parasite's next host is a crab, so they increase their predation rates. But the final host are birds, migratory birds. And so it goes from birds to snails. And so what happens is birds love mudflats. So as these die-offs increase over time, the snail fronts, is that these predators that aren't snail predators, they're eating worms and crabs, come in and they fly in and they parachute through their poop a lot of parasites. And that we find higher infection rates on there. So and it slows down the front. It actually has a big effect. So very cool question about that dynamic. Thank you. I like it.